God, we look to you tonight. Above the singing, God, above the music, above the songs, let your name be lifted high, Jesus. Let your name be honored, God. And we commit all that we are to you, God, and everything that we do. And we reach towards you, Jesus.
Down on my knees again, surrendering all, and find me here, Lord, as you draw me near. I'm desperate for you, I'm desperate for you.
First conference Society of Nigeria, Day 3, virtual presentations. We are about to start the presentations now. Please, if you are online and you are ready to present, please, you will signify. First person we have on the list.
The first presenter this morning is Damfami Abigail. After Damfami Abigail, followed by Ania Dike Ezon Melu Maria. Yeah, I want to call three so that they get ready and line up. And then the third presenter will be Bawa Saidu. So the first presenter is Damfami Abigail.
Hello, Damfami, are you there? Damfami Abigail, are you there? Hello, online. Is Damfami online? Abigail. Okay, next person.
Next person is Maria. Anyadike Ezonwu Melu Maria.
Is Maria online? Maria.
Yes, Prof. I'm all right.
Okay, Anyadike. Is it Maria Anyadike?
Yes.
Okay.
Sir.
Now, the floor is yours. You have 7 minutes to present and 3 minutes to respond.
Okay, sir.
Share your files. Okay, go ahead. Your time is going.
Okay, good morning, everyone. Good morning, Prof. and my fellow colleagues. I'm here to present a conference paper on conservation status of trees in Nnamdi Azikiwe University. Introduction. Trees play a vital role. Trees plays a vital role in both ecosystem and human life, providing numerous benefits that contribute to the health and well-being of our planet and its inhabitants. In ecosystem, trees serve as a foundation supporting a wide range of plant and animal species through provision of habitat, maintaining soil fertility, preventing erosion, regulating the climate, maintaining air and water quality, aesthetic uses, and cultural and spiritual significance. By conserving and protecting tree populations, we can help maintain the health of our ecosystem and support the well-being of human societies. Tree conservation refers to the practice of protecting and preserving trees and their habitat to ensure their survival for future generations. It is also referred to the sustainable management and protection of tree species and forest ecosystems to ensure their existence and the provision of ecological, economic, and social benefits. There are various methods of tree conservation which include reforestation, afforestation, sustainable forestry, maintaining a healthy and diverse forest ecosystem. Threats faced by trees that can impact their health, growth, and survival in the ecosystem include deforestation, urbanization, invasive species, pests, and diseases. So it is important to reorganize and address these threats in order to protect the health and survival of trees and the ecosystem. Aims and objective. The aim of the study is to find out conservation status of trees in Nnamdi Azikiwe University, Awka. The objective of these studies include understanding the ecological importance of trees and their roles in ecosystem, to survey and determine the tree species composition, diversity, abundance, and dominance, identify and assess the threat to tree species and their habitats, develop the strategies to protect and preserve trees in their ecosystem. This is the study area showing the map of Awka and the Nauru University, that's Nnamdi Azikiwe University, Awka. Methodology. The study assessed the conservation status of trees in two study areas. One of the study areas is the Botanical Garden of the university and the University Beautiful Gate site. So the survey study is a survey study, and the study area is visited. Investigation was thoroughly conducted using a stable and convenient sampling method. One plot is 30 by 30 meter and is randomly laid on transect so that we can get the compositions. Each of the trees found in each plot were identified to species level with their botanical name and family. The result. Table 1 shows the composition of trees in botanical garden. Here it shows that the tree species at the botanical garden where the Tremontosa shows the tree with the highest frequency of 6, followed by the Cleistophoris patterns and others, which are 2 and 1 frequency. Then at the Beautiful Gate, the University Beautiful Gate, we have Terminaria catapa to be the highest composition trees, followed by the Geminella arborea and Acacia auriformis. Then the species, the diameter breadth height at the botanical garden, it shows that the Pentacletra macrophylla has the highest dBH, followed by the Copper abaca and Kanga odorata having the lowest dBH. Then the tree height, Elias guinnessii, that's the palm tree, has the highest tree height, followed by the Pentacletra macrophylla. Then at the University Beautiful Gate, we have the Geminella arborea to be the tree with the highest diameter breadth height, followed by the Terminaria catapa and with the lowest tree height and dBH, while Geminella arborea has the highest tree height and the crown cover and dBH, which is diameter and breadth height. Then species diversity at the botanical garden, we have the Terminaria tomatosa to also be the tree species with the highest diversity, abundance, and dominance, followed by the Cleistophoris patterns, just as the species composition. Then their families, from the study, the Utricaceae has the highest species under their families, followed by the Fabaceae, Euphorbiaceae, and the lowest includes those with one frequency. Then at the University Beautiful Gate, the three species, the family with the highest tree diversity is the Combretaceae and Laminaceae, while the Fabaceae has the lowest species diversity. Then conclusion, the differences in the number of the tree species recorded in the sampled plot, that's the sites, may be due to variation in ecological factors and other habitat conditions which favor more tree growth, diversity, and distribution. At the Botanical Garden, the most frequently occurred and dominant tree species was under the family Eutricaceae, which could be attributed to the fact that their seeds are dispersed by wind. They have rapid regenerative capacity connected with symbiotic attributes, which might have facilitated the species to effortlessly start to exist in available ecosystem categories. In comparing the two study sites, it revealed that the study area have that the Botanical Garden has it revealed that these two study areas have different dominant species, that their species composition and diversity are different. Terminaria catappa has significantly larger crown cover at the Botanical Garden. When you compare the Terminaria tree species at the Botanical Garden and the one at the University Beautiful Gate, it shows that the one at the Botanical Garden has larger crown cover than the one at the University Beautiful Gate, which could be as a result of age or space competition.
You just have a minute left.
Okay, sir. The University Beautiful Gate have limited species diversity and smaller tree size, which indicate the vulnerability of the tree to more human activities, while the Botanical Garden have diverse and mature tree population, which suggest a more stable ecosystem and could be as a result of over-exploitation and anthropogenic activities. Thank you for listening.
Okay. Please, a round of applause for that beautiful presentation. Thank you, Mary. Let me just say right away that your tables, go to any of them, their headings appear incomplete. Hello, Mary, are you there?
Yes, Prof.
You have stopped sharing your slide.
Okay.
Okay, just take note of that. Your tables,
Okay.
the headings are not complete in most cases.
Okay, Prof.
Do we have any question from the audience here?
I could see from your tree listing, though they are tiny for reception to the eye, you need to edit that tree list. Despite the fact that it's tiny, I could realize that your Kananga odorata is not correct. It's not well spelled out. It's Cananga. Then also Pricralima nitida, not well spread out at all. It's supposed to be capital P, small I, Cralaima. Thank you.
Okay, noted, Prof.
All right.
Thank you, Maria. What is the size of your quadrat? I want to know the size of your quadrat.
Hello, sir. Is it the quadrat I used during the sampling?
Yes. You should know. Because the sampling method will determine your result. I had her saying that she used 40 meters by 40 meters. So if you use 40 meters by 40 meters for sampling trees, you will have a very small quadrat, and there could be no informative information coming from that type of quadrant. So if you are sampling trees, you need like 200 meters by 200 meters. If you are sampling herbaceous plants, grasses, this way you can have 5 meters by 5 meters. So the size or the growth form of your plants determines the size of your quadrant. That's my observation. But I heard her saying 40 by 40.
Mary, are you there?
Yes, sir.
Okay. I'm particularly concerned that you've mentioned a lot of invasive species and their importance, value are significant. Example, Melina arborea. But you seem not to actually talk about that in your discussion. It's important. It's a concern because we are having invasive alien weeds and species invading our ecosystem. So if you actually, in a university, an institution that you have that, you should actually have recommendations that they should look at using indigenous tree species than introducing these ones that are potentially damaging to the ecosystem. Okay?
Okay, sir.
So you review your discussion and recommendations to capture that. Thank you.
Thank you, sir.
All right, Mary or Maria, thank you for your presentation. You can
Thank you, sir.
step down now. So the
Thank
next
you.
person, sorry to say this right now. Sorry to say this at this moment. We are reviewing down because of the number of people that have signified online. The timing to 5 minutes, please. It's 5 minutes now you have for the presentation. Okay? We'll still maintain that 3 minutes for discussion afterward. So please, the next presenter, are you ready? Hello?
The next presenter is Bawa Seidu. Just go straight to your point, and I think 5 minutes should be able to summarize.
Bawa Seidu, are you there? Bawa Seidu. Bawa Seidu. Bawa Seidu is not seated. Hello? Okay.
The next presenter is Taiwo O Ayodele. Taiwo. Then the
Yes,
next
sir.
one is Sani
Taiwo
Seidu.
Ayodele.
Yeah, after Taiwo is Sani Seidu Bago Biri.
So it's Taiwo now. Taiwo Ayodele.
Yes, Taiwo Ayodele is active, sir.
Okay. Sani Bagobiri, be ready. Okay. So Taiwo, you have the floor now. Please be reminded, it's 5 minutes. Okay?
All right, sir.
All right.
Thank you.
Yeah, you can take off.
All right. All right. Sorry. Okay. Good morning, everyone. I am Taiwo Ayodele, Science and Technology, Ikarekiti, and I will be talking on the effects of beauty effluent on morphology and leaf anatomy of Celosia argentea. Here is my outline. Introduction. Celosia argentea is a genus of edible and ornamental plants which belongs to the family Amarantaceae. And among the different species of this plant, Celosia argentea is an important leafy vegetable of high nutritional value. Industrial effluent have great inhibitory effects on germination and development of many plant species. And during beer preparation, wastewater, that is the effluent, is mainly generated by pressing wet grains. And also, beery effluent carrying abundance of organic carbon, and plants were found to alter the soil chemical properties. Objectives of the study. The objective is to, one, identify the effect of effluent treatment soil on the growth of Celosia argentea, two, to determine the effect of effluent treatment soil on the leaf morphology of Celosia argentea, and finally to examine the effect of effluent treatment soil on the leaf anatomy of Celosia argentea. Materials and methods. This experiment was carried out in the screenhouse situated at the Department of Botany, Obafemi Awolowo University, Osun State, and the collection of samples. Sandy loam soil was collected from an abandoned site in Modakeke, Osun State, Nigeria, while the Celosia argentea seed were sourced from National Horticultural Research Institute in Ibadan, and the brewery effluent for the treatment was obtained from International Brewery in Ilesha, Osun State, Nigeria, for the experiment. Now to the experimental design. A moderate-sized container was filled with the soil, while Celosia argentea seeds were sown and irrigated. Also, after 14 days of planting, the Celosia seedlings were transplanted into 25 bags and allowed to stabilize for further 7 days and irrigated throughout the process. At regular interval, from a day to the treatment, growth parameters of morphological features such as shoot height, that is from the base of the plant to the terminal bud, the leaf length from the base of the leaf to the apex, and the leaf breadth, the distance of the widest portion of the leaves, were measured for all controlled and treatment samples using metric roller. Now for the anatomical analysis, Celosia argentea the anatomical analysis of Celosia argentea leaves was carried out on the controlled plant after 4 weeks of planting and 2 weeks of exposure to brewery effluent. After 6 weeks, some leaves were harvested for epidermal peeling. While observing the epidermal peeling, epidermal layer of both adixia and abaxia leaf was obtained by soaking the leaves in water, carefully scraping with blade to expose the epidermal layer, and also mounting on a clean slide inside glycerol after staining with Safrani O to make the stomata structures visible, and then sealed with nail varnish. And then the slide were further observed under magnification 400 to observe the stomata type, distribution, epidermal cells. Results. The results on the shoot height showed that the height of the treatment plant from the figure, it shows that the height of the treatment plant was significantly higher than those of the controlled for the first 9 days, while a reverse scenario was observed from day 11 to 13th of the experiment, whereby the treatment plant showed significant reduction as compared to the control. Also on the leaf area, it can be seen that there is a significant difference between the treated plant and the controlled plant. And initially, the controlled plant showed a significant reduction compared to the treatment for the first 9 days. However, from day 10 to 13, the controlled plant showed a progressive increase, while the treatment plant showed a significant reduction. On the adixia surface of the celosia, with respect to the stomata frequency, the length and breadth, it can be deduced, it can be seen that there is no significant difference between the controlled and the polluted plant. The same result applies to the abaxia. Discussion. The brewery effluent had effects on the morphological characters such as reduction in shoot height, leaf area, and biomass of treated Celosia argentea plants. Statistical analysis of the vegetative growth showed that there is significant difference in all the vegetative parameters studied. The results in this study showed that brewery effluent restrained the growth of plants as evidenced by reduction in shoot height. This finding corresponds with the data presented for Tanilium triangulare for launch until 2016. Also, the decrease in leaf area reported under 30 ml effluent concentration could be noted as the effect of effluent on plant in concentration. This is in agreement with Ogunwemo et al. 2010, who stated that brewery effluent affected percentage and rate of germination of Celosia argentea. Conclusion. Celosia argentea in this case respond to pollution of brewery effluent with initial increase in its morphological features, which later reduced towards the end of the study. Also, the anatomical features appear not affected at all. These attribute the pitch that the plant has capacity to survive the challenging environment. And finally, by leveraging on the unique adaptive mechanism of plant Celosia argentea, we can develop effective strategy for pollution mitigation and ecological restoration. Recommendation: additional studies should be conducted to explore the long-term effects of bio-effluent pollution on the anatomical characters of the plant Celosia argentea. Thank you for listening. Hello.
Thank you, Tayo, for the presentation. The floor is open for questions.
Thank you, Tayo. Your work is thorough and beautiful. But I have this to ask. In your conclusion, you said that the anatomical parameters were not affected at all.
Yes, sir.
But you told us that in the morphology, that the early growth morphology revealed that the effluent actually significantly affected growth, right? The parameters. So
Yes.
it is expected that an increased leaf area should correspond with an at least increased number of stomata. So,
Okay, at the early stage of the experiment,
hold on.
right?
Yes, so how do you marry this? Because it is known plants with broader leaves should actually also have a wider or a higher distribution of stomata. So how come that the stomatal difference, that difference is not actually captured in the stomata? How can you justify that? Thank you, sir.
Okay, thank you. That's because the observation was made at the last stage of the experiment. So necessary changes has been implemented on the plants during the treatment. Did I answer the question, sir? Hello?
Hello.
Yes,
Hello.
sir. Yes, I'm with you, sir.
From what he actually said, did you measure stomata size? If you never did,
Yes.
you should. But you are not showing any record that has to do with stomata size. Because I wanted to say that if you never did, you just must do. You don't have any chart signifying that you actually did stomata size measurements.
It's in my thesis.
So are you trying to say that from your thesis, the stomata sizes were equal? There was no differentials. Is that what you are trying to say?
Yes, sir.
I don't think so.
So please take these observations into consideration before you move on to the next stage with your work. Thank you very much.
Yes,
The
thank you,
next
sir.
presenter is Sani Saidu Bagobiri. Sani Saidu Bagobiri. Are you there?
Yeah, actually,
Is that Sani Saidu? Okay, we move to the next presenter. Amubiya Omolara Faith. Amubiya Omolara Faith. Pardon my pronunciation of these names. You know, sometimes they sound like botanical names, so.
No problem, sir.
So.
Good morning, sir.
Good morning. The floor is yours. Share your slides.
Okay. All right, sir. Good morning, my prof.
Morning.
Thanks, sir. Okay. Is my slide visible now, sir? Okay.
Share your screen. It's not.
Is my screen visible now, sir?
This is something else, but it's your screen.
Okay.
Maybe you close the Word file and move to your PowerPoint.
All right, sir.
Get ready, Amujiri Angela N. and Murjatu O Musa. Can we have you, Faith, or we move to the next person?
Yes, sir.
Are you ready now?
Yes, sir.
Okay, go ahead. Share your screen, your PowerPoint.
Okay.
Yeah, it's coming up.
Oh, thank you very much, sir.
Put it
Good morning, sir, my prof, sir.
Put it on presentation mode. The screen icon, yes. At the task bar, the bottom, that red line, the screen bar is there, icon is there. So click on it.
Okay. Which one, sir?
At the bottom of your screen, of your PowerPoint, there is presentation mode on that red line. Almost to the last, yes, there.
Yes, sir. Thank you very much, sir.
Hopefully it will come up.
Is it coming up, sir?
It's not. It's loading.
Okay. Sorry about that, sir.
So you have 5 minutes to present your points.
All right, sir. My name is Amubiaya Omolara from the University of Ilorin. My topic is a synergistic role of nanoparticles and avuscular mycorrhizal fungi in boosting polycyclic aromatic hydrocarbon uptake in 2 phytoremediator species. This is my outline. Environmental pollution is increasing gradually and causing serious impact on living organisms, including human. Nigeria, being a developing country, is one of the nations in the world that is being faced with the challenges of diverse forms of pollution. And one of our major environmental challenges is soil pollution caused by oil spillage, especially in the oil-producing part of the nation. In the last 5 decades, Nigeria has suffered environmental degradation as a result of oil pollution. Oil spillage results in the contamination of the environment with aliphatic and aromatic hydrocarbons. Among the aromatic fraction, polycyclic aromatic hydrocarbons are given prime attention as they tend to pose toxic effects on the environment and as well as human. The aim and objective of my study, was to assess the impact of avuscular mycorrhizal fungi and nanoparticles and the combination of two in phytoremediation of crude oil contaminated soil. And the objective is to evaluate the agronomic performance of the model plant, which is a Vetiver cicinoides and Senna occidentalis. Then, and also to evaluate the effect of AMF, avuscular mycorrhizal fungus, which is Glomus clarum, and cerium oxide nanoparticles, and the combination of these two on the growth performance of these two plants, and also to evaluate the pH uptake under different treatments. This is my materials and method. The potted experiment was self-taught in an improvised greenhouse, and avuscular mycorrhizal fungus was sourced from IITA, Oyo State. Bony light crude oil was obtained from Nigeria National Petroleum, Port Harcourt, and Vetiver root was sourced from a village in Ondo State, while cerium oxide nanoparticles was obtained dry from Melorium Technologies, UK. So then the soil was spiked, the sandy soil was bulk and homogenized to give one representative. After 2 weeks of, after I've sieved it, 2 grams of the soil were weighed per pot, and each was spiked with 2 ml of crude oil homogeneously mixed together and left for 2 weeks to agglomerate. Then seedling were now transferred from the nursery bed into each pot that contained 5 seedlings. At transplanting stage, 20 grams of glomus clarum was blended into the central third of the soil. Also, 200 milligrams of serum oxide nanoparticles was also added to the pot. So this is the treatment. This is how the treatment was arranged. Soil grown with Senna occidentalis with 20 mils of crude oil. Soil grown with Vetiveria cicinoides with 20 mil of crude oil. That is the control. And then soil with all this, with Vetiveria cicinoides plus 20 mil of crude oil and 20 gram of glomus clarum. With soil grown with Vetiveria cicinoides, 20 mil of crude oil, 20 mil of nanoparticles, and then this is my result, the characterization of the serum oxide nanoparticle. This shows that the serum oxide nanoparticle is in its pure state, as shown in the EDX analysis. And this is the Fourier infrared spectrum, where I analyze for the functional group present in these nanoparticles. And then this is the result for the percentage uptake of PAH in the soil. I have 2 plants combined together under different treatment. The sample A represent Vetiveria cicinoides and sample B represent Senna occidentalis. Then in the result, it was shown that Vetiveria cicinoides, Senna occidentalis actually outperform Vetiveria cicinoides in the uptake of PAH. But nevertheless, the addition of serum oxide nanoparticles and AMF treatment proved to uptake more of PAH across all treatments when compared to the control soil, which was solely polluted with crude oil without any form of without adding AMF or nanoparticles. The growth parameters also shows that soil amended with AMF in both Senna occidentalis and Vetiveria cicinoides outperformed the soil polluted soil. And also for the height of the plant decreased significantly in the crude oil polluted soil, which could mean that crude oil contamination significantly impaired the growth and the development of these 2 plants. And then the uptake of polycyclic aromatic hydrocarbon. Now, in the soil polluted soil treatment, both Senna and Vetiveria cicinoides exhibited limited uptake of polycyclic hydrocarbon in a soil polluted soil when compared to the treated soil. Now, the introduction of arbuscular mycorrhizal fungus and nanoparticles notably improved PAH uptake in both plant species under combined treatment of polluted soil with AMF and nanoparticle. Senna occidentalis actually demonstrated 100% uptake for several PAH, such as benzopyrene, and then while Vetiveria cicinoides exhibits are lower compared to Senna occidentalis. In conclusion, the results of this study indicate that both NP and AMF positively influence the agronomic parameter of Senna occidentalis, Vetiveria cicinoides in crude oil polluted soil conditions. Similarly, results of the PAH uptake indicate that the combination of this of AMF and nanoparticles, serum oxide nanoparticle, has significant potential in enhancing the phytoremediation of crude oil contaminated soil. Thank you.
Okay, thank you for this presentation.
Thank you, sir.
Agronomic parameters of Senna occidentalis. Can you expatiate on that? Is Senna a crop?
Senna is a plant, sir. The common name is we call a Senna coffee.
Is it a crop?
No, sir.
Okay, okay. So, how useful will this be trying to understand its agronomic parameters?
Hello, sir.
How useful is trying to understand its agronomic parameters if it is not a crop?
Okay, because for it is being used for phytoremediation purpose, and then any plant that serve as a good phytoremediator must be able to grow well on contaminated soil. That was the reason I assessed for the growth parameter and to see how well this particular plant can do when grown on contaminated sites.
So if senna is not a crop, that means it's a weed.
Yes, sir.
Okay, so why not its ecological parameters? Is it possible instead of agronomic? You know, there are different perspectives.
Okay.
What have you got to say to that?
Well,
Okay.
I will.
You will think about it.
I will look into that, sir,
Okay.
because it's not one of my objectives, sir. So
Okay,
I will.
okay.
Thank you for the suggestion, sir.
Yeah. Any other question?
You see, our point is that when you say agronomic, is a Greek. Agriculture is cultivation for commercial purposes. So that's why we're emphasizing why not ecological parameter.
Ecological, all right.
After all, the vetifera also, maybe insect killer also, or what have you, natural insect killer, is not a crop also. So that's our own contention.
All right. Thank you very much, sir. I will look into that, sir.
Yes.
And do the necessary correction, sir.
Yeah, please, to add to that, you go to your discussion, you buttress your discussion by telling us the mechanism in which the arbuscular mycorrhiza enhance uptake of this contaminant. Is it by increase in root length? Okay. Or does it make it absorption easy? Or you understand? Let the mechanism yes, let us know the mechanism.
Yes, sir. I actually have a report for the root length.
Exactly.
The arbuscular mycorrhiza fungus.
Yes.
But I didn't use it for this particular presentation. The particular treatment that contained the arbuscular mycorrhiza fungus actually increased the size of the root length, sir. It buttressed the spreading of the root as well.
But you have mentioned it here, so it should also be in your discussion and in the work, because you have mentioned it. It's here in your ears, so please put that in this presentation.
All right, sir. I will do that, sir. Thank you very much, sir.
Our online audience should be able to have time to ask questions as well, so we'll give you the opportunity. And then for Faith, I think for next time, if you want to present, it will be wise to use the image of the plant so that people will not ask you, what is this Senna occidentalis? And I like the idea that you're using this plant because I've seen it growing on west side. If there's any other plant is growing, especially Senna, you will always see it. So it's good that you have tested this and see how Senna is coping with the west side ecological niche. So it's a good work. So let's give online audience the opportunity to ask if they have questions.
Any question from online? Online, please feel free to interject and call our attention so that we can give you audience. Studio, maybe you show us the online in case somebody is raising his hand. On share, Faith, on share your slide.
Thank you, sir.
We want to check if anybody in the audience online. Online, do you have any question? You can signify by raising hand so we can give you space. Okay, there is no question, so thank you for your presentation, Faith.
Thank you, Prof. Sir.
Yeah.
Our next presenter is Amujiri Angela N. Amujiri Angela N. Are you there? After that is Murjatu O Musa. But the floor is for Amujiri Angela N. Murjatu is what I see here.
Yes, Mujatu is ready.
Sorry, say that again.
Mujatu is here and she's ready.
Okay, so the floor is yours. Can you share your slides?
Okay, thank you, sir.
The next presenter is Hussaini Aminatu Adam. Hussaini Aminatu Adam, Ajayi Isaac Isuolua.
Please, can you see my screen now?
Yes, so but put it on presentation mode. Perfect. Go ahead. You have 5 minutes to present and 3 minutes for comments.
Okay, thank you, sir. Good morning, everyone. I'm Mujatu Musa from the Department of Biological Sciences, Abubakar Tafawa Balewa University, Bauchi. And I'll be presenting on the paper, Ethnobotanical Survey and Abundant Studies of Azanza Gakena in Tulawangi, Gombe State. Azanza Gakena is locally known as Gorontula, and it's a multi-purpose semi-domesticated shrub that is valued for its medicinal, nutritional, and ecological significance. Despite its widespread use, there is limited documentation to its ecological status and traditional applications, especially in Tulawangi. And the objective of this study is to determine the traditional uses and socio-economic roles of Azanza Gakena, to determine its abundance and conservation status, and to collect and prepare virtual specimens of the plant. This is the fruit of the shrub Azanza. The one on my left is the unripe fruit with the leaf, and the one on my right is the ripe and harvested fruit. And this is also a picture of the tree. The description of the study area. The study was carried out in Tulawangi, which is located in Katungo local government area. The area lies within the Sudan Savannah, and it is socioeconomically diverse, with inhabitants ranging from farmers to civil servants, and it has basic infrastructure, which include educational institutions, healthcare facilities, which contribute to the improved standard of living. And here is a map of the area. My methodology, the sample size for this study was 40 households, and it was selected based on the projection of the 2026 population survey using this formula. And for my data collection on the ethnobotanical data, I conducted structured interviews and focus group discussions with herbalists, farmers, and elders of the community. For the abundance analysis, I laid 15 quadrants measuring 50 by 15 meters randomly to check for the following indices: the relative density, dominance, frequency, and important value index. For my results, the ethnobotanical survey reveals the extensive use of this plant in Tulawangi. From the survey, the fruit is a most utilized plant, and is primarily administered by chewy and it's widely used as an aphrodisiac to enhance fertility, as an antihypertensive and and for diabetes. And over 70% of the responses emphasize the reproductive health benefits. Other plant parts have been mentioned, and they said it's been used to treat other ailments like ulcers, malaria, piles, and other metabolic disorders. Beyond the uses for medicinal plants, it's also used for some traditional purposes like ash making. Some of them utilize um, burnt ash for shampoo and even as an antiseptic. For the results of my abundance study, 16 species were identified. Azaza Rakena had the highest IBI, is 401.35, and the highest density and relative frequency. Sorry, relative density, not density, and relative frequency. And it was followed by Adansonia dubicata, which had the highest basal area, and also Pachia biglobosa. Other species like Azadrapta indica and Pardavia albida also recorded moderate representation, and they had least relative dominance of species like Prateva.
You just have 1 minute left.
Okay. So my conclusion, Azaza Rakena is an ethnomedicinal plant. It is the most ecologically dominant species nowadays. Its multifunctional value underscores its relevance to local health and livelihood system. Despite current pressures, it's remains concerned on the IUCN list. And recommendation, I recommend that the sustainable harvesting should be promoted in communities, and they should validate the pharmacological properties of all parts of this plant. We should encourage awareness to preserve both the species and indigenous knowledge. Thank you very much for listening.
Okay, thank you. Thank you for your presentation.
Thank you, sir.
Okay. Any
Thank you, Mujatu, for the presentation.
Thank you, sir.
I expect that you should be conversant with the current trend on the plants that you are studying. So Azanza garciana is no longer called Azanza garciana. It has been moved to Thespecia. So it's now called Thespecia garciana. Are you aware of that?
Okay, based on my own observations,
T-H-E-S, Thespecia.
Thespecia garciana, it's still listed as Azanza in the IUCN list, but the Thespecia was mentioned as synonym.
The popular name is Azanza. I agree with that. But I'm just telling you that the name has changed based on molecular phylogenetic studies by some group team. They changed the Azanza garciana and is moved now to Thespecia because it's no longer fit into Azanza. And I want to guess that your plants, the one that you are studying here, is the one that is in Tula, Gombe State. And if it is the one that is in Gombe State, it's no longer in Azanza garciana. That is what I want to tell you. It has been moved to Thespecia. And then, it was considered to be endemic to Gombe.
Yes, I'm listening.
But some species or some individuals of this Thespecia, they are now found somewhere else, like especially in the Eastern Africa. So review and be updated with your literature on the plants. Secondly, I see you are working on ethnobotany of Azanza, as you call it. But I've observed that you are using...
Hello, sir.
You are using...
Hello?
Hello,
Yes, I can hear you now, sir.
can you hear us?
Yes, I can hear now.
Okay, so I'm saying that your work is on ethnobotany of Azanza Garkiana. That's the topic. And in the main work,
Yes, sir.
I've seen that you're working on diversity of plants of Tula, maybe, because you're talking about frequency, important value index, and so on, which to me is no longer only Azanza. For you to be able to get IVI and other frequencies of the plants, you must have studied. Yeah, so in that context, it's no longer only ethnobotany. It's now ecological studies. So there's a mix-up in your work. And then from your quadrant, you used 40 by 40. I can imagine that.
50 by 50.
Sorry? Or 50 by 50. Still is too much.
Yes,
it's too
sir.
small for plants, for trees. Okay? It has to be more. It has to be like at least 100 meters. At least 100 meters for woody plants. Okay, so can you clarify what you did? Is it ethnobotany or ecology, the diversity of plants or woody plants in that study area?
Ethnobotany and the abundant study of the plants.
Okay. Just think about these posers and do your best to upgrade your information, especially on the plant and its new taxonomic status. Is that clear?
All right, thank you very much, sir.
Okay, thank you.
Yes, sir, it's noted.
Okay. Thank you. Who is next?
Husseini Aminatu Adam. Husseini.
Yes, sir.
Okay, the floor is yours. Share your slide. As she's sharing her slide, the next after Husseini Aminatu is Ajayi Isaac Isiolua. After Ajayi is Dana Zumi Ebanma. Okay, the floor is yours. Aminatu.
All right, sir. Thank you. Good morning. I'm Hussein Aminatu Adam from Department of Botany, Faculty of Life Science, Ahmadu Bello University, Zaria. And I'm here to present a work on impact of climate change on species distribution and ecological niche of Paikia species in the West African sub-region. Introduction. The genus Paikia, named after Mongapart in 1826, belongs to the Fabaceae family of the flowering plants. The primary product of this species, known as Obula or the dawa, is a highly valued spice in Africa. It is anticipated that greenhouse emissions will have a major impact on the distribution of plants across the world, and these changes will likely have impact on the growth, production, and distribution of native plant species in Africa. Ecological niche distribution model make use of digital maps of the environmental variables and spatial information on the vegetation factors, species, and its abundance. It is a methodology that is often used to predict how much species distribution respond to climatic differences. The aim of this study is to forecast the impact of climate change on spatial distribution of Paikia species at present and in the future scenarios under African 4.10 and 8.5 Horizon 2070. Materials and method. The study area for this study is the 16 countries of West Africa, and West Africa is on latitude 4 degree and 28 degree north of the equator, and the longitude 15 degree east and 60 degree west. Here is a map of West Africa showcasing the 16 countries. This study made use of two major data sets, which includes the occurrence data and environmental variables. And the occurrence data for this study were gotten from GBIF, and the environmental variables for the current we downloaded from WordClimb, and for the future environmental variables, that's ICP 4.5 and 8.5, were gotten from AfriClimb. And this is a flowchart for the methodology. After getting our data from GBIF, we cleaned the data using QGIS, and we extracted our environmental variables and format them to ASCII format to make it compatible for the max endpoint. We evaluated our model using 30% of our occurrence data for validation, and we use 10,000 as maximum number of background points, and also remove duplicates. Data analysis. The selection of models was done using threshold independent tests. The impact of climate change on species distribution were extracted based on threshold, and the change analysis was done using the Monix model on QGIS. Results and discussion. Figure 1 shows the ROC and AUC pop of species model at present, which gave us an accuracy of 0.61, and the AUC accuracy test ranges between 0 to 1, with values closer to 1, giving us an information on how well our model is. Figure 2 shows the spatial distribution of Pachia species as present, and here our result shows us that most part of northern West Africa is not suitable for the occurrence of Pachia species, though the southern part of West Africa shows a promising species suitability habitat, with most of the central part of western West Africa has a highly suitable habitat for Pachia species. Figure 3 shows the predicted distribution of Pachia species under ICP 4.5 Horizon 2070. Here, there is a noticeable shrink in habitat suitability of Pachia species as compared to that of the present. We have
You have a minute. Just 1 minute left.
All right, sir. And the impact of climate change on Pachia species in present condition on ICP 4.5. There's a little change on areas that are earlier unsuitable and thought suitable under Africa ICP 4.5. And the next slide shows the impact of climate change on Pachia species between present condition and ICP 8.5. Here, there is a more noticeable change on habitat suitability that are earlier run unsuitable and later turned suitable. Impact of climate change on Pachia species between ICP 4.5 and ICP 8.5. It shows the same image as before, with little areas that are earlier unsuitable turning suitable. Here, the impact of climate change, this table gives us the impact of climate change on Pachia species across West Africa.
Your time is up.
All right, sir.
Your time is up.
So, conclusion, the results suggest that the substantial portion of West Africa are unsuitable for Pachia species under both scenarios. 0.89 to 1.69 of West Africa is predicted to transition from unsuitable to suitable under these scenarios, and 7.23 to 7.79% of West Africa is predicted to transition from suitable to unsuitable under these scenarios. The recommendation for this acknowledgement. Here are my selected references. Thank you for listening.
Okay. Thank you. Thank you. Thank you for your presentation. Okay.
Thank you very much for the presentation.
Thank you, sir.
You said 4 species in tropical West Africa. You never. You said we have 4 species from your listing, distributed throughout the tropics, with 4 species found in Africa. No.
Yes, sir.
4 species in Africa.
Yes, sir.
So
4 species in
throughout the
Africa.
whole world. 4 species in Africa. Then you never told us how many species in tropical West Africa. That one is implied. and how many species in Nigeria. And out of those 4 species, either in Africa, tropical West Africa, and Nigeria, you never had a table listing the species.
Yes, sir, I have them on my thesis.
Okay.
But I didn't see a table list.
You can assuage our doubt by mentioning them. That you
All right, the
have,
4 species found in Africa include Paikia biglibosa,
okay.
Paikia filae, Paikia Madagascariensis, that's from Madagascar, and then Paikia bicola. Those are the 4 species found on the African continent.
Okay, okay. Thank you. Thank you.
You're welcome.
Thank you for the presentation.
Thank you, sir.
How many climatic variables did you use?
17, sir. 15, sorry, 15.
15, they are too much. It causes what is called effect of collinearity. So by that we mean one factor can serve the same purpose with another factor. So there's a way you will remove factors that are contributing the same thing to your modeling. So if you do a threshold of their correlation, you will cut off those variables that will contribute the same with the other thing so that you avoid the overheating of your model that will give you false information. So using 15 variables is way too much. That's first observation. The second observation is that some of the...
So I later reduced them to 4.
You reduce the variables to 4?
Yes, sir.
Yeah, then that should be your factors. It's reasonable,
okay, sir,
but
yes,
4
sir.
is even small. And then second observation is that you should have added soil data. So instead of using the 15 variables that are just climatic, you can add soil data, you can add elevation, because these are also factors that are driving species distribution.
Yes, sir.
Another thing that I did not see here is that from your map, I've seen that northeast doesn't have much occurrence data of Paikia. But if you look at Bauchi Gombe, going into Adamawa, Taraba State, you will see that I think the dominant species of trees in this area is Paikia, biglibosa. But clearly from your map, maybe you have deficiency in your sampling. It's not indicated. So I suggest you have a thorough search of your data, and if you don't have, then you can outsource them. Otherwise, you will give us a false distribution of these species. And given that you are handling only Paikia, I can suggest that you can do all the species across the globe and model them. It's not difficult to do. You can use the same model, the same formula, the same script to do for the rest of the species globally, instead of restricting yourself to West Africa when you can achieve the global distribution with the same script.
Yes, sir.
So
yes,
I don't
sir.
know which package you are using, but if you are using the one suggested by Philip, then that will be the best approach.
Yes, Maxent.
Maxent, yes, yes. So you can use R to model all the species across the globe and you get a better view. And then another doubt that I have is that this is just one species, because one species is responding to the model different from another species. So if you have done for 4 species, I expect 4 maps of West Africa with each species showing how they are responding. Because if you go to northeast, like I mentioned, the one I know very well, Paikia biglibosa is responding to its environment or to its ecological niche different from Paikia speciosa. So I expect that you have 4 maps, each for one species, and showing how each species is responding to the factors that is driving their distribution.
Okay, sir.
All right.
Noted.
Okay, okay. I'm just wondering, what made you pick on Pakia? Because Pakia is already one of our enduring plants in the field. It's one of the strongest enduring plants. So is this a worst case scenario you are trying to paint? What's the reason for picking on Pakia?
All right, thank you for your question, sir. Yes, though Pakia are currently not on the red list, but they are plant species that are of high value to our community. And its primary product, which is the dawa, is made from its seeds. And there is high overuse of these seeds rather than for them to be used for regeneration for next generation of Pakia. They are used for the dawa. So there is a noticeable change in reduction of young Pakia that I imagine. So I did pick Pakia to check how well would they adapt to climate change, as most of the occurrence that we are having now are aging.
Okay, okay. Thank you. Thank you for. Okay, there's no more question for you. So thank you for your beautiful presentation.
Thank you, sir.
Our next presenter is Ajayi Isaac Isiolua. Ajayi Isaac. You can unshare your screen, Aminatu. The floor is for Ajayi Isaac. After Ajayi is Dan Azumi Ebanma. Is that Ajayi on the floor? Yes. You have 5 minutes to present your points quickly. Don't dwell on the methodology. And tell us the objectives, your results, and what you think about the work. Then we will engage you for 3 minutes discussion. Share your screen on presentation mode. Can you hear us? Ajayi, can you hear us? Hello, can you wave to us if you can hear us? You cannot hear us. He has left the place. Are you ready?
Yes, Prof. Hello, Prof. I can hear you.
Yeah, go ahead. The floor is yours.
Oh, okay. So, I said my topic, paper presentation will be based on hybrid production, studying of genetic diversity and gene expression for early maturing and delay ripening traits in tomato. No, tomato belongs to the family Solanaceae and is one of the model plants for plant breeding. The statement of problem, there has been low production of tomato in Nigeria as compared to other continents like Asia, American, and the like. And one of the major challenges facing tomato processing factory in Nigeria include delivery of fresh tomato fruit with desirable traits, such as the delay ripening and the early maturing traits, which has often led to deterioration in production. So this study is to enhance yield, to check how we can enhance yield by targeting early maturing and for increasing productivity and as well as delay fruit ripening in tomato. And also, this study we ensured or has ensured the integration of variants in production rather than just maintaining the genetic stability in transferring genetic material from parent to offspring. And also, there is a need to improve tomato production by using transcriptional genes, which are responsible for these traits. That's the early maturing and delay ripening that is being targeted. So the aim, as I said, is to produce hybrids through crossing, as well as assess genetic diversity among tomato ascension collected, and also to detect transcriptional gene associated with the early maturing and delayed fruit ripening in tomato. So the objective to the characterization of quantitative and qualitative traits, to hybridize our parent accession in order to evaluate early fruit ripening, delay ripening, early fruit maturing, delay ripening and yield. Also to evaluate ploidy level, that's the mitotic chromosome of both the parent and the F1 progeny, to determine genetic diversity using the SSR marker, and as well as to evaluate to determine the expression of the gene associated with early ripening and delay using SLIARF gene, that's the Solanase transcriptional gene. So the materials and method: phenotypic assessment was done, morphological was done, genetic assessment using SSR marker. 8 primers were used for this study. These are the list of the primers. Then these are the list of the gene. The first 3 genes were used for the early maturing traits, while the other 2 were used for the delay ripening. The early 3 rather was used for delay ripening, while the other 2, the 4 and 5, were used for early maturing, while the 6th were used for as a control. So the morphological data were collected as well and were analyzed. So from the results, 47 ascensions of tomato were collected from various germplasm, including NIOT, NABGRAB in Ibadan, and also from the market in Ekiti State. And they were characterized for different traits as shown in the table display. So then crossing was done based on the phenotypic assessment of those traits, based on the characterized traits of interest, whereby the late ripening tomato were crossed with early ripening, and as well as early maturing were crossed with delay ripening, in order to have a hybrid which are characterized for the traits of interest. This table shows the quantitative traits in which 18 quantitative traits were characterized, and the morphological shows high level of variation across the ascension that were collected. And also the quantitative character as well, the performance of the table 8 shows the performance of the tomato fruits from the parent, and it was observed that both for the fruits, yield, and agronomic character, it shows high level of variation. And this also helps to give a better information about the parent ascension that were used for the crossing, because the parent ascension were ascensions that shows high level of that shows better characters, and as well those ones that shows a poor performance when they were characterized. So this one also shows for the F2, where the F1 hybrid was collected from the cross. The hybrid 1, 2, 3, 4, 5 were product of the parent cross, and they shows high level of variation among the fruits and the yield traits. Then the tomato were stored, and this is the information. This was what informed the delay ripening ascensions, whereby the ascensions were subjected to the same method as described in the methodology for storage, and it was observed that the rate of deterioration varies across the ascensions, whereby, as for example, the ascension NHTO were crossed with the NGP 00713, and the hybrid that was produced, which is hybrid 1, shows an increase in the rate of deterioration, and this helps to analyze this helps to better characterize the ascensions, and as well as help to know the level of the level of relatedness across the ascensions. Then the correlation analysis, the PCA, the principal component analysis was done. Then also among the ascensions that were crossed together, both for the hybrid and as well as they were crossed together to show
Just
the
a
level of
minute.
the relation. Then these are the
You have one
This is
more
the
minute. One more minute.
okay. For the discussion, the key character shows that plants' height, number of inflorescence, and other morphological character varies across ascension. Then this finding shows that the mitotic, the crossing, helps to increase variability, and this was also confirmed by using of the SSR marker that were used for the diversity study. Then across those ascensions, the ascensions that were crossed for early maturing and for delay ripening, the product, which are the hybrid, shows a level of significance in terms of gene expression, in which it was observed that there were expression of the early maturing gene and the delay ripening gene in those tomato ascensions. So then the conclusion, there are some ascensions that were used for this study shows and also some primers like the SSR 47, 63, 268 exhibit some level of distinct fixtures which can be exploited for breeding purpose, as well as the ring gene and other gene that were used were also were expressed for morphological traits as well as for fruit firmness, for delay ripening, and as well as number of days to maturing for the early maturing traits. So thank you.
Okay. Thank you for your presentation. Thank you. Thank you so much. I was lost at a point. I thought I heard you say mycotic.
No, I said mitotic chrome. I had a crack of voice, so my voice was not too clear.
Yeah. I later realized you were referring to mitotic. Okay. At times, we need to give it an extra effort to ensure our diction and pronunciation for the sake of the audience. Is that clear?
Okay, Prof.
Yeah.
Okay, Prof.
Okay.
You said you evaluated ploidy levels and mitotic chromosomes, and you eventually showed us some photomicrographs. So which technique did you use going about your mitotic chromosomes?
I use the snatch procedure.
What is it about, the snatch? The snatch procedure, what is it about? Is it about staining and the usual?
Yeah, it's about staining and visualization. Staining and visualization.
Okay.
Any other person?
The next presenter is Abigail Amfami.
Is there anyone online that want to ask him a question? Anyone online? Unshare yourself for now. Let's see if somebody online wants to ask a question. Okay. Anybody online? Okay, thank you very much for your presentation, Ajayi, I believe.
Thank you, Prof.
Thank you.
For our next presenters, please take notes of this. We want to see the evidence of your payment, your membership renewal payment. So you tell us whether you have paid and the date you paid so that we can check before you present. Our next presenter is, we called the name before, but she's ready now. Amfami Abigail. Are you online? Ajayi, you can, on share? Oh, done for me. Okay. So the floor is yours. You have 5 minutes to present your result, and we will discuss it for 3 minutes. Share your slide and put it on presentation mode.
Can you all see my slide now?
Put your slide on presentation mode. The screen icon at the taskbar. Yes. Are you there? After Abigail, the next presenter is Danazumi Ibama. Danazumi Ibama.
Can you all see my slide now?
No, we cannot see.
Are you seeing my slide? I want to start presenting.
We cannot see. Share it.
Can you? I have shared it.
Yeah, we cannot see. It's not here. You shared it before and you stopped it. You shared it before, you stopped it because you wanted to put it on presentation mode. She's getting ready, so the next presenter is Danazumi Ibama. Are you online? Danazumi Ibama. After that is Abdullahi M. Bello. We are saying this so that you get ready. If one is not ready, you can present. Damfami, are you there?
I'm here, sir.
Yes, so share your screen. Yes, she has paid. Yes, she did. With evidence. Now it's coming up. Okay.
Okay.
You unshare again.
You can now see my screen, sir?
No, you have unshared it just now. Okay, the next presenter is Abdullahi Angazumi Ibama. Are you there? Angazumi. Abdullahi Mbello. Is Abdullahi Mbello online? Abdullahi Mbello. Dr. IU Karaye. Karaye? Karaye. Karaye. Are you there? Dr. IU, are you there? Karaye. Amfami, are you there?
Sir, yes, I'm here. I can hear you, but
Share your
shared
screen.
my screen. So you...
Share your screen. Share it.
Can you see my screen now?
No, we cannot. You are not sharing.
I've shared it. Maybe it's the network.
But the network will not give us the voice if you share. If it is network.
Yes, sir.
Okay.
Can you see it now?
No.
What of now?
Yeah, so we'll just skip on.
Can you see my screen now, sir?
No. So the next is Amfami
What of
Ebaba.
now?
So go
Can you
ahead,
see it now?
even if it is like this.
Okay. Good morning, everybody. All protocol duly observed. I am Danfami Abigail, a PhD student from the Department of Plant Science and Biotechnology, University of Jos. This morning, I'm presenting on the topic yield and yield component of some land races of and wild relative of Bambara groundnut grown in two savanna agroecologies in Nigeria. The study is aimed at determining the yield and the yield component of some Bambara groundnut species and their wild relative grown in two agroecologies in Nigeria. Aim. The study is aimed at determining the yield of the Bambara groundnut and their wild relative at different agroecology, and also determine the yield attribute that can be used in plant improvement of Bambara groundnut. Introduction. Bambara groundnut to the height of 30 to 35 centimeter in height. The stems grow it have a creeping stem on the ground level where the leaves grow directly from the stem. Bambara groundnut have nutritional value. In spite of its nutritional value, no much research attention was given to the plants, so it has been classified as one of the orphan crops we have. Then Bambara groundnut also is used in some African countries like Nigeria and Ghana in making some traditional delicacies. Also, Bambara groundnut, apart from the nutritional value it has also medicinal value. The black variety of Bambara groundnut is being grounded into powder and mixed with guinea fowl meat in Ghana to cure diarrhea in children. The red variety, which have high level of protein, is also used where iron deficiency is high. Materials and method. The germplasm of Bambara groundnut were collected in different agricultural development programs in Nigeria, as well as the wild variety being collected from Cameroon Republic, from hunters in Kunji in Cameroon Republic. Some of the variety of the Bambara groundnut that was used in this study were collected from local farmers, and some are collected from gamers in Todi, Biliri local government area of Gombe State. Experimental site. The research was carried out in two different locations, one in Dadinkowa, Gombe State, and the other in Kuru, Plateau State. The Dadinkowa location is fall within the Sahel Savannah, while the Kuru location fall in the dried savanna. Experimental design. The experiment was laid in a completely randomized design with 4 replications. Each replication is measuring 3 by 2.4 meters, while the gross plot size is 66 by 0.5 meter by 14 meter. Cultural practices. The plot mapping, harrowing and tilting was done manually, and also plot mapping. The seed were planted at interrow and inter-row spacing of 0.8 and 0.2 respectively. Weeding was done at 4th and 9th week. By the 10th week, etching up was done to cover the young pods. Harvesting was done at 146 days after sowing, when the leaves turn yellow and also the plants begin to, the leaves begin to fall off.
You have 1 minute.
Okay.
Go to what you did. Tell us your result. What is the take-home message?
The result. From the results, the data that were collected, first one was the final stand count at harvest. All the parameters that were determined show significant, all the varieties shows, the accession shows significant difference. And also the performance of Dainkowa was higher than Kuru in all seasons, both 2022 and 2023. And also, that is in all the parameters that were under this study, from the final stand count, number of seed per pot, shelling percentage, number of pot per plant, and total grain yield. Then also the heritability, genotypic coefficient of variability, phenotypic coefficient of variability, heritability, and genetic advance as percentage of mean were also studied. And also in this study, both years and both seasons, they showed moderate to high phenotypic coefficient of variability and genotypic coefficient of variability, and also heritability and genetic advance as percentage of mean population, according to Jonasson in 1949, who invented the formula, and was also reviewed by Demfami and Namu in 2021. Then also, in conclusion, all these parameters, the result of this study shows that the parameters that have moderate to high genotypic and phenotypic coefficient of variability can be used as selection indices in improving of Bambara groundnut. These parameters are final stand count at harvest, number of pot per plant, numbers of seed per pot, and also...
Your time is up. Thank you, Abigail, for the presentation. Before I give the audience the floor, I would like to clarify. I would like you to clarify us on what's the botanical name of the plants you are studying. Is expected
Vigna subterranea.
Yeah, it's expected that you speak botanical language, because as she didn't. Yes. So and then the difference between Bambara groundnut and Bambara nuts, we don't know. So we need to know this. And then next time, it's advisable that you present, especially the first slide where you introduce the concept. We should be able to see the pictures of your plant so that we can validate whether you are telling us, or what you are telling us is about the plant we see. So someone who does not know Bambara groundnut will be able to see the picture. And you have mentioned varieties. We needed to see the varieties. Okay, so these are few observations. So Bambara groundnut is a common name, and we know the implication of the use of common names as botanists. It's complicated and it's confusing. So you should have used simple scientific name for us, so that if you are presenting it to the ministries or farmers, then you can use Bambara groundnut or Bambara nuts, as the case may be. The floor is open for questions. Any question?
You mentioned Dainkowa. When you mentioned Dainkowa, you're supposed to put the state. Then the Kuru, you're supposed to put... let me just say what I want to say first. Then the Kuru, you're supposed to put the state too. Because it seems you are emphasizing Nasarawa, Plateau, and probably even Gombe State or so. Then you are mentioning some farmers in Cameroon. So maybe you can defend those statements.
Okay. Okay. The first location is Dainkowa in Gombe State, Yamatu Deba's local government of Gombe State. Then that is the one that falls in the Sahel Savannah agroecology. Then Kuru in Plateau State, the one that falls in dry Savannah agroecology. Then also, you made mention of Nasarawa. Yes, I collected the cream variety with white eye from Nasarawa Agricultural Development Area. Then also the wild variety of Bambara groundnut. That there are two wild varieties that were used in this particular study. We collected it from Kunji, from a hunter in Kunji, a Republic of Cameroon.
Your affiliation is University of Jos, not so?
Yes, sir.
Okay. Are you aware that There is a place called Adinkowa in Jos.
Yes, sir.
And there is also a place called Nasarawa in Jos. Huh?
I don't know, sir.
Nasarawa
I don't know, sir.
Oh, okay. Okay. Now you can imagine the level of confusion you have created. You know? Because incidentally, the terminology for your points of collection are all located in Jos. So always give details that will put doubt to rest. There is Nasarawa in Jos.
Okay,
There
sir.
is all these other places also in Jos. Okay?
Okay, sir.
All right.
Yes, sir.
Thank you very much for
Thank
your
you so much, sir.
presentation. Thank you. Next.
Next is Angazumi Ibama. You can unshare your screen, Abigail.
Thank you, sir.
Ibama. After Dangazumi is Abdullahi M. Bello. We don't know whether he's online, so we are waiting for him to respond first. Unshare your screen, Abigail. Dang Azmi Obama, are you there? Abdullahi M. Bello. Any of these two online? Next is Dr. Ayu Karaye. Karaye. Okay. Abubakar Shehu. Is Abubakar Shehu online? Are you there? Nwanko Onyebuchi E. Nwanko Ebuchi E. Subair Bolariwa Khadija. Amujiri. Saidu Bagobiri. Are you ready? Saidu Bagobiri. Okay. Kajidat.
Yes, sir, I'm ready. Saidu Bagobiri is here. I'm ready,
now.
sir.
Okay, it's your third time now. Have you paid for membership renewal or
Yes,
registration?
please. I have paid, sir.
When?
Yes, sir.
When did you pay?
Yesterday, I paid for the membership and
Prof.
I paid for registration on
Prof.
22nd.
Payment. Bogobiri yesterday. Yes.
Yes, sir.
The name is Sani Seidu Bogobiri.
Yes, sir.
Okay, the floor is yours.
Okay, sir. Let me share my screen. Yes, sir. I'm here.
After Bogobiri is Khadija Suleiman Jafar.
Sir, please, can you all see my screen?
Yeah, we can see your screen. It's your time now. 5
Yes,
minutes.
good morning, sir, and
Present
good morning,
your work,
everyone.
5 minutes, 3 minutes for discussion.
Okay, sir. I am Sani Saidu Bagobiri from the most peaceful university in the country, Usman Danfodio University, and I am going to present my paper, Improving Sorghum Drought Tolerance Through Phosphate Solubilizing Microorganisms. Sorghum is considered as a versatile cereal crop deeply rooted in the West Africa, and it is considered by many authors that it is a food that can be used to address global hunger challenge we are facing. And then also, sorghum is a sorghum is belongs to the family Poaceae, and then drought is one of the challenges that is causing sorghum yield loss in Nigeria. And according to this scholar at BIU, it is reported that it is due to the short rainy season, which lasts for 3 to 4 months. On the other hand, there are many researchers that discover that phosphate solubilizing microorganisms can be used to to control drought stress in plants, or to improve plants to be drought tolerant, because drought tolerance can be caused by lack of nutrients. So in this research, the paper aims to investigate it aims to improve drought tolerance through phosphate solubilizing microorganisms. The study was carried out in the physiology laboratory of the Department of Plant Science in Usman Danfodio University. And also the plant materials were collected in the SADP, Sokoto State, where a variety was collected to serve as the drought-sensitive variety, which is Samsung 45, and then another variety was used, a local variety, which was brought to the herbarium for identification, was used as a drought-sensitive variety. It has no any prior treatment. And also culture of Bacillus subtilis, Bacillus lentus, and Aspergillus niger were collected from the department from a previous study conducted in the year 2021 in the department. The experimental design, full factorial design was used because I have for 3 factors with different levels. So I decided to use I'm having network issue here. And also for drought, I have 2 drought levels, which are 15% drought stress and 20% drought stress. I represented them as 15 and 20. And for microorganisms, I have Aspergillus niger. Also, variety, and I have 2 control groups, which were made after the factorial regression. The design control groups, which are positive and negative control. Positive and drought-sensitive variety that has been conducting the experiment. I osmoprine my seed with polyethylene glycol, which is an osmolyte that mimics drought stress.
1 minute.
And also seed planting. Seeds were planted in a pot.
You have 1 minute.
Okay, sir. So the flow chart shows my methodology, and then I measured 17 parameters. 9 were pigments using CID VIU machine. And then this is the result. The highest chlorophyll total was obtained in treatment 3, and then the chlorophyll content index was obtained in treatment 10. And this is a chart that shows the result. Treatment 3 appears to have the highest chlorophyll content, and also this is the result for other pigments related indices. And this is the table that shows the result there. And this is the growth parameters. I measured germination percentage, seedling vigor index, relative water content, shoot length, root length, fresh weight, and dry weight. And this is the result for germination percentage, seedling vigor index, and related water content. And this is in conclusion, seed germination and seedling growth of sorghum, sorghum bicolor may be aided by seed priming and bioinoculation using the soil drenching method as in Shiva Stava, according to the other studies which support this theory. The best performing treatment appeared to be treatment 3, which contains a certain organism. That means it performed better than the control, even though one of the control performed, but
Thank you, Bagobiri. Thank you for your presentation.
finally, this approach can be used. Yes, sir.
Thank you. Finally, next year. Um, I will advise our presenters to concentrate on your findings. In these 5 minutes, you can tell us the objectives of the work and your findings. Probably our take-home message. Instead of spending 4 minutes on methodology that we know. You can mention in your results, for example, you can say, in this quadrant, we know that you use quadrant for that sampling. And then you tell us what you find in that quadrant. That way you will save your time and you'll be able to pass your message to us. Okay, so
I hope you can still be hearing us. You are still hearing us. Hello. Maybe he has left. You are still hearing.
Hello.
You are still hearing.
Sir, my network is.
But you can hear me now.
You can hear me now. Yes, I can hear you, sir.
In your introduction.
Yes, sir.
You said in Nigeria. There is problem of drought resistance because of the rains lasting for some 3 to 4 months. So I think it's better you rather put it there that in its major areas of production. I hope you understand what I'm trying to say.
Noted, yes, sir.
So that it doesn't seem as if we generalize that it's only 3 to 4 months rain months in Nigeria. So in its major areas of production. So it balances it better.
Yes, sir. Thank you, sir.
Okay, thank you. Thank you for your presentation.
Prof, sir.
Okay.
Thank you for your presentation. It was really wonderful. But then, I saw that in your analysis, you mentioned that you use a regression, a type of regression, but you have not seen that in your inference. Because I believe that the regression was supposed an interaction between the drought tolerance or drought sensitivity, the cultivars, and the other factors in the experiment. But it's not in your inference. We've not seen it. And you've neither told us about the accuracy of the prediction model you actually derived from this work. So you looked that up. You tried to highlight that. Because that should actually be the selling point for this presentation. Thank you.
All right, you can stand down. Okay. Who is next? Who is ready from those that were called earlier? Who is ready? Hello? Yes. Who is ready again? Signify if you are ready. Is Angela on seat? Amujuri Angela. Amujuri Angela. Unmute yourself so we can hear you. Amujuri Angela.
I'm going back to your work.
Okay, Amujuri, we can hear you now.
Okay.
Uh-huh.
Can I share my slide?
Yes, share your file, and then continue.
The aspect of
Share your file.
How can I share my file? So you go to a site, they gave a very good descriptions and the statement on that. You have to infuse it into your methodology. Okay. You write that describe the research design and
Who is speaking?
I'm trying to share it, please. Sound very, very well.
I think we will come back to you. We will come back to you.
I have started sharing it.
Take your time. Don't take your time.
Now, can you just summarize for me?
We will come back to you.
In 2 sentences. Okay. All the wood extractive you carried out.
Do we have Dan Azumi on seat? Do we have Dan Azumi on seat? What of Abdullahi Bello? Abdullahi Bello. Abdullahi Bello. Next. Dr. Karaye. Do we have Karaye on ground? IU Karaye.
Hello?
Do we have Abubakar Shehu on ground?
Abubakar Shehu is on ground.
Abubakar Shehu. Okay, share your. Okay. Have you paid membership dues? Abubakar.
Yes, I have paid yesterday. Yes, I paid, sir, yesterday.
Okay, how much?
Hello, sir.
Yes, how much?
Hello?
Yeah.
I paid 20,000 for bike shot. Hello, sir.
Hello, can you hear us?
Yes, I can hear you, ma'am.
That's for conference registration. Have you paid your membership due?
No, I haven't paid my.
You have to pay that before you can pay.
Hello? Hello, ma.
Yes, the procedures, you have to pay the membership due before you can present. Is Amujuri Angela available now?
No, Dr. Karai. Dr. Karai is available now.
Dr. IU Karai.
Hello, sir.
Yes.
Yes, please. Yes. I'm trying to get you, please.
Is Dr.
I'm trying to share the. Yes, available, please.
Dr. IU Karai. Are you available?
Yes, please. I'm ready, please.
Okay, share your.
Yes, I am. Inshallah.
Have you paid your membership?
Yes, I paid until 16th. Until 16th, please. Hello. Hello.
Have you paid your membership due?
Yes, I paid all. I paid everything.
How
Yes.
much? How much?
Hello.
We are hearing you. Have you paid your membership
Is
due?
it? Yes, I paid. I paid. I paid, sir.
How much?
10,000. 26 June.
Yeah. 6 June. Okay.
26 June. Yes, 26 June. 26 June.
Yeah. Is it as new member?
Yes, yes, as new member.
6 June.
No,
No. There's nothing like that. Is it 26 or 6?
What?
What did you say? 26 or 6? Let's check. Just check 26 if you can find it.
I've checked. I'm sure he paid for conference registration. It's not here.
Who is
Hello?
this? Amojuri. Okay. Amojuri, we can see your file, but have you paid your membership? Can you hear us?
Dr. Kara is trying to share his screen with you.
Please, to all our online participants, the procedure is that you have to pay the annual membership due to qualify you to present. So, Amujuri, we can see your file. Are you hearing us? Amujuri, are you hearing us? Please, studio, unmute so that change the kini.
Okay.
She's not responding. She's not responding. So let us please change to another person. Studio, assist us. Focus on another person. Studio.
Doctor. Hello. Membership. Oh, ba member ne kyau.
Amujuri.
Ba member ne kyau.
Unmute yourself, Amujuri. Unmute yourself.
I have unmuted myself.
Okay, we are hearing you now.
Okay.
Now, wait. Have you paid your membership?
Yes, I have paid my membership.
When?
No, I don't pay for, but I paid for conference fee and the preparation of processing of abstracts.
But the membership due, you have not paid.
Yes, I have never been there. I didn't register as a member.
Oh.
I registered as a participant.
Okay.
My party is low.
Okay, Amujuri.
Yes.
Share your file.
Yes.
Share your file. Share your file.
Okay, my battery is becoming low. I'm coming. Let me plug it.
Then the strength of the bamboo increases.
They said that I should share my file. Okay.
Amujuri, she's out. Her battery has gone down. Okay, who is next? Abubakar, okay. Next person. Call another name. Call another name. Do we have Wanko Oyebuchi on ground? Wanko Oyebuchi. Wanko Oyebuchi. Wanko Oyebuchi. Wanko Oyebuchi. Please, if you are online.
Please, this is Dr. Garai, please. Ready, please. Dr. Garai, yes.
Yes, we are ready to receive you, Dr. Karai, but you are not answering our question.
I have answered sorry, what's the question?
The question is,
yes,
have you paid your annual dues?
Yes, I paid everything, sir. I paid.
How much and which date?
Total 35,000. 35,000, please.
Sorry, please. You pay 10,000 for conference due.
Yes.
For conference registration, right?
Yes.
Are you a PG student?
It's another person. No, I'm not PG student.
You are a staff?
Yeah, I'm academic staff, yes.
Okay, how much did you pay for conference registration?
25,000. 25,000.
25. But
yes.
Your name is not on our list for membership. 10,000. You said 6th of June?
26, not 6. 26, please.
We can't find your name.
Yes. 26 of June.
Okay, share your file. Share your file.
Okay.
We are not seeing the file yet. Okay. Okay. It's coming. We have not seen your slide.
I'm trying to.
Okay, Karai, we can see your slide. You have 5 minutes to present.
Thank you very much, sir.
Your time starts now.
All right, sir. Thank you very much, sir. Thank you very much, sir. This is Karai Ayu from Udus Sokoto. All protocols please duly observed. I'm here to present this on the effect of lead on seed germination and early growth of millet, that is Penicillium glaucum. Next slide. On management activities such as livestock grazing, farming activities, mining activities result in environmental degradation. Fossil fuel combustion, flooding, mining, and eutrophication further deteriorate the environment. It is evident environmental degradation adversely affects seed germination, growth, as well as the biodiversity of plants in general. Metals are of special interest with respect to the toxicological importance to human health, plants, and animals. The lead is one of the most common heavy metals contaminants in soil. Plants are extremely harmful to it. Lead has little or no biological purpose in plant, but can create morphological, physiological, and biochemical problems. Aim and objective of the study. The aim of the study was to determine the effects of lead on seed germination and early seedling growth of millet, that is Penicillium glaucum, while the objective of the study were to determine the effect of lead on persistent seed germination as well as the germination rate. Second is to assess the effect of lead on growth parameters such as shoot and root lengths as well as the number of leaves. The study area. The experiment was carried out in the Department of Herbarium, Department of Science, Usman Affan University, Sokoto, main campus in Sokoto. The Sokoto lies between latitude 10 degree 10, 0.5 north, 2.5502 west, and 5 degrees east. Is that? Both the nursery and plantings were conducted in the same botanical garden. Seed collection and authentication. The seeds of the species in question, that is the millet, were procured at the Sokoto Agricultural Development Project, Sokoto. The seed were taken to the Battle Herbarium for authentication and issuance of Herbarium voucher, Herbarium number, sorry. Voucher specimens were equally deposited there. Baptism method include the germination rate was assigned according to the method as described by Gallup et al. 2022. Parasitic germination was determined following the method of ISTA 1995. The results, you can see from the tables, the treatments in milligram per kilogram, and germination percentage, germination rate, root length, shoot length, and number of leaves. You can see from the control, that's number 1 there, the percentage was up to 96.13%, while the rate was 0.81, root length was 7.21, shoot length was 6.21, and the number of leaves were 14. This is the next treatment, that is 50. It dropped to 75.92, and germination rate was 0.56. Root length dropped to 54.43, and shoot length also for 3.12, while the number of leaves dropped to 10. You can see that proceed up to the level 300, where the percentage germination were just 18.32%. So you can see that exactly. So the control group showed that highest germination percentage, that's than 6.13%, while increasing the lead concentration led to the significant reduction down to 18.32 at lead 300. This aligns with the previous studies showing that impairs seed germination, disrupting enzyme activity, and altering membrane integrity. Both root and shoot lengths decrease significantly with increasing the levels of the lead. Is that from 7.21 to centimeters to 56.21 centimeter in control to 8.21 centimeters and 5.65 centimeters also, respectively. This actually indicates that the lead inhibited elongation and divisions of the cell. Is that disrupt water and nutrient uptake and damages meristematic tissues. Yes. To conclude, it is evident that lead contamination has dose-dependent inhibitory effect on the millet seed germination and early seedling growth. Long-term exposure may also affect the crop yield as well as the other physiological activities. It is recommended that phytoremediation using the plants to extract the lead is highly important there. A number of species have been reported to have this phytoremediation ability. The next one is the soil amendment using bicola, biochar, lime, as well as addition to the yes. So that's it, please. Thank you very much. Thank you for reviewing. Hello.
All right, Doc, thank you very much for your beautiful presentation. Okay.
Thank you very much, sir.
I want to ask,
yes,
is this a real practical scenario?
Yes.
In your area, the incidence of lead?
Yes. Quite, you know, Sokoto, neighbor to Zamfara states, which have been battling with the issue of mining for this gold there, rampantly doing there, exactly. And also, this is, I've just said that emission of these petroleum products and other human activities led to this contamination, please. Only that the scenario is not that pronounced compared to some preference exactly here. But for the crop in question, that is the millet, is one of the major cereal crops in Sokoto. So that's why that's called for the
Yeah, I can appreciate it now. I appreciate.
Yes.
I hope you can still hear us. You are still hearing us.
I'm still hearing.
Please, pertaining to your title.
Yes.
Maybe I talking to you as a taxonomist.
Yes.
That title, after that millet, I would rather prefer that. After that millet, if you can have an iPhone or a dot or what have you, the contention is that L there is Linus, is lean.
Linus, yes.
So
Linus, yes.
And that RB is Robert Brown.
Yes.
So if you can remove,
Robert Brown, yes.
If you can remove those two brackets and put L in bracket, then after that millet, maybe you can have two dots or an hyphen.
Okay.
So you can't just have lean Robert Brown. Lean worked first before Robert Brown. So it should be in bracket, that lean.
Yes. So thank you very much.
Yeah, just cross-check on that.
Okay. Inshallah.
Okay. Do we have any other query for him? Okay. Only that I think on slide number 3,
yes.
I see something about, is that 3?
3?
Is that slide 3?
Slide 3?
Slide number 3.
Number 3, yes.
No, this number 4. Abi? Just step the slide backward a bit. 1.
Okay. 1.
Okay, 1. 2. Wait. Okay, wait. Yeah, it's something that look contrary. About lead
yes.
Being toxic to the plant, or the plant being toxic to the lead. Yeah, actually.
It's the lead that is toxic to the plants.
Next slide. Maybe we'll cite something.
Okay.
So anyway, please just check. There's one of the slides that sounded like that. Thank you very much.
So thank you very much.
Yeah.
Sorry, please, my brother. I have a brother, please. Abubakar Shehu.
Okay.
We are together, please. He's ready to present, please.
Okay.
Abubakar Shehu.
Thank you very much for your presentation.
My brother, please, Abubakar Shehu. We are together, seated here, please.
Okay.
Yeah,
so you can unmute now. Thank you for your good presentation.
Yes, the Amijir is ready. Amijir is ready.
Okay.
Please, Abubakar Shehu.
Amijir.
Please.
Yes.
Please unshare yourself, Dr. Karai. Unshare yourself. Unshare. Thank you. Amujiri.
Yes.
Yeah. Go ahead. Amujiri.
Yes, I'm trying to share it again.
Okay, go ahead. Share. Okay, go ahead. No. Shehu Karaye, please. Unshare yourself. You cannot share until we tell you, please. Thank you. Okay, Amujiri. Amujiri.
Sa Abubakar Shehu. It's ready.
Hello. Hello. Please, they told me to share my surprise. Hello. Hello.
Okay, Amujiri, go
Yes.
ahead, go ahead. You have 5 minutes.
Okay. I'm Ujiri AN. My topic is influence of downside of on floral abundance and heavy metal composition in Enugu State. As locations for waste disposal, downsides have been linked to environmental degradation because waste material interacting with the surrounding environment poses long-term consequences such as soil contamination, air pollution, and the destruction of local ecosystem. Though, with the passage of time, plant life colonized landfill, even with the difficulties of its soil toxicity and instability, a number of plant species root in these unwelcoming locations. Those sites serve as major repository for municipal waste in the cities. They also act as potential epicenter for plant invasion which affect local ecosystems and biodiversity. Interestingly, recent studies show those sites dual purposes to mitigate environmental impacts associated with dumps like methane while providing renewable energy sources. Understanding the plant distribution pattern in dump site is crucial for effective waste management, invasive species control, potential bioenergy production, soil improvement, reducing erosion and mitigating greenhouse gas. Studies observe that certain plant species have adapted to the extreme conditions of landfill sites, such as contamination from heavy metals. Hence, species capable of tolerating contaminants are essential for effective land rehabilitation. Moreover, the role of plant communities in landfill ecosystem has broader implications for ecological restoration strategies, as emphasized that the long-term success of landfill reclamation require an integrated approach, combining plant life with soil amendment and careful monitoring. Therefore, the research focused on the influence of downside on abundance and heavy metal composition. That is the aim of this study. Materials and method. A study was conducted on three wisdom sites on Enugu State, Nigeria, the Abakwanike Site A, UNEC Site B, and the Ugbene 1 Site C. 5 soil samples from 0 to 30 cm depth were collected, bulked together, mixed as composite samples, and bagged for physical-chemical analysis. Estimation of flora used standard modified contract method according to Botanical 2013 to obtain percentage frequency for floristic diversity. Heavy metal concentration, that is cadmium, arsenic, lead, nickel, and zinc were assessed in the soil and edible plant sample using atomic absorption spectrophotometry. Results. The results of soil physicochemical show that highest exchangeable gases are seen in type A, highest pH and organic content as are obtained in type B, and electrical conductivity, temperature, organic matter, and and nitrogen are highest in site C. pH results also show that pH range from slightly acidic to alkaline. Organic matter range was from 2.86% to 3.40% with the highest organic content carbon content inside B and very low electrical conductivity was also seen across the sites. Also, site A has higher soil metal, cadmium, zinc, arsenic, lead, and magnesium.
You have 1 minute more.
Okay, this is the table. This is the table for soil heavy metal, and the total of 31 plant species, including 13 edible belonging to 16 families, were recorded with the following percentages presented here. That is the percentage frequency of occurrence we obtained across the dump site. Luvas lindrica, followed by Alternaria brasiliana, were the most abundant plant species, while Convolvulus was the most abundant family obtained. Ipoma batata and Amaranthus varieties were the most edible plant species that absorb heavy metals, and all of them are mainly abundant in site A and site C. And
Okay.
this is the
Thank you. Thank you. Your time is up.
no,
Thank you for your beautiful presentation. Let me first of all refer you to your title. Your title. Please, may I ask, are you an ecologist?
yes, yes,
You are an ecologist.
yes.
Okay, okay. Because flora, as in your title,
Uh,
and flora.
those it's not supposed to have the L. Please.
Okay, beautiful,
Not
beautiful.
supposed to have the L.
Yeah, thank you. You have assuaged our doubts and confusion. You have cleared many things. Thank you so
Okay.
much. Thank you so much.
Okay.
So you will remove the L, then
Yes, yes.
people will understand it better. Okay,
Okay.
okay. Let me also know uh, how many dump sites did you survey?
I surveyed 3 dump sites.
Okay, okay,
Abakpa, Abakpa UNEC campus, and Ubene 1.
okay, okay. Did you have any data
Yes.
is insinuating on any particular differences, either in nature or structure of these dump sites?
The the data, I don't have particular data like that, but the type of waste being dumped there is what we thought may differ in the frequency of occurrence of plants and even accumulated and the heavy metal abundance.
Okay, okay. So the nature of the dump
Yes.
influences the flora
The
that you
yeah,
find in the
floor,
place.
percentage occurrence of flora.
Okay, 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 okay. Thank you so much for your informative presentation. Thank you.
Thank you.
Yeah. I've looked around here. Nobody has any question after you cleared that big doubt. Thank you
Okay.
for your presentation.
Thank you.
So you can
Thank
step down now. Unshare yourself.
Okay.
Thank you.
Thank you.
Thank you. Okay. Who do we have on ground? Who is ready? Yes.
Hello.
Ona Chingadi Deborah.
Who? Uh, Who is on ground? Who is ready? That's
Honor Deborah. Honor Chimdiadi
okay.
Deborah.
Honor. Have
Yes,
you
sir.
paid the necessary fees, the membership?
I'm an undergraduate, sir. I paid the registration fee.
Okay. 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 This is Dan Azumi we are seeing. Please don't share yourself until we say so. Dan Azumi, please unshare yourself. Thank you. Okay. So, Ona.
Yes, sir.
Okay.
Yes, sir.
Share yourself.
Okay, sir. Okay.
Yeah, you have 5 minutes. Your time is
Can you see it, sir?
on. Yes.
Okay, sir. Okay, good day. So I'm Onachimdadi Deborah, and we worked on the anti-inflammatory properties of
Put
the
your
methanol root
file
extract.
on presentation mode. It's too tiny for us to see here. Presentation mode.
Okay.
Okay.
Can you see it now, sir?
Good, good. Go ahead.
Okay. So we worked on the anti-inflammatory properties of the methanol root extract of Combretum morianum excel and the family of Combretaceae. So Combretum morianum is a plant that is native to Sierra Leone. So and it's in the family of Combretaceae, like I said earlier, and there have been various works that have been published about this Combretum species for their antioxidant properties and their anti-ulcer properties. So we worked on the anti-inflammatory properties. We got the proximate composition and then went further to do the animal studies for the anti-inflammatory properties. So the mechanism of inflammation, inflammation is the body's defense mechanism against detrimental external stimuli. So the mechanism of inflammation, like I listed here, includes the recognition where cell surface receptors recognizes the detrimental stimuli and then activates inflammatory pathways, leading to the release of inflammatory markers like the inflammatory cytokines. So for my materials, I listed them here. We made use of the distilled water. We use distilled water for control as well, for the negative control. And then we used diclofenac, which is a well-known non-steroidal anti-inflammatory drug for the positive control. And for the anti-inflammatory property, we use the acute model, which is the carrageenan-induced rat paw edema. And we used formalin-induced rat paw edema for the subchronic model to evaluate the anti-inflammatory activity. So from these results, the percentage, our plant extracts had a good yield. It was in line with what was in the guideline. And we carried out an acute toxicity test, of course, on the rats, but none of the rats died at the doses we carried out. We carried them out in 10, 100, and 1,000 milligram per kg doses for 3 rats each. So after we didn't see after we didn't observe any mortality, we went further to 1,600, 2,900, and 5,000 milligram per kg doses, and we didn't observe any deaths. So there was no LD50 calculated. So we concluded that our plant extract is non-toxic. And this is the chart showing the percentage inhibition of inflammation for the acute toxicity, where we got the percentage inhibition of inflammation over time. So, and this is the one for the percentage inhibition of inflammation over days. So we did for for this one over time, we did for 30 minutes and then we did for 1 to 5 hours. Then for the days, we checked for 1 to 6 days. Rather. And our results showed that for the acute inflammation study, our plant extract was active at the highest dose, which was 400 milligram per kg. So after 2 hours at the highest dose, it recorded a very good percentage inhibition of inflammation compared to the standard drug, diclofenac. And for the subchronic inflammation study, our plant extract at 400 milligram per kg dose also recorded a high percentage inhibition of inflammation at the on the fifth day rather. So we concluded that the methanol root extract of Combretum murianum excel plants from the family Combretaceae is good enough. It demonstrated good anti-inflammatory activity. So it supports the traditional use of these plants in treatment of inflammation. So instead of using non-steroidal anti-inflammatory drugs that lead to gastrointestinal side effects. Thank you very much, sir.
Thank you for your beautiful presentation and for keeping to time. All right. Can you show us the figure you presented? Yes. Please bring it back to presentation mode. Okay. The figure.
Yes, sir.
Okay. I'm just trying to look at the footnote because this is not a title now. Is it a title?
No, sir.
The footnote there says chart showing the percentage inhibition of inflammation. So how do we read that percentage inhibition? Is it vertically?
Yes, sir, vertically.
Meaning what?
It was labeled percentage inhibition of inflammation is vertical, while the time is horizontal.
So interpret it.
And
interpret it as the taller the what, and the shorter the what.
Yes, the taller. Okay. The shorter ones.
I think she's supposed to give figures, not taller. The lines are supposed to represent values. I can see 40, 30 there. So
yes,
the values that she's supposed to give. Even though the key there enters into the value there, is supposed to be interpreted based on values, not taller. I think.
Beautiful.
So she's supposed to give the figures for the values, sir.
Beautiful. But you now agree with us here that, presenting it like this can lead to misinterpretation. Yeah. And
yes, sir.
So you make the necessary adjustments so that people can understand it when they see it. All right. Okay, thank you.
Okay, sir. Thank you, sir.
I hope you are still hearing. Hello.
Yes, sir. Yes, sir. I heard you, sir.
From your write-up, you wrote that the plant is native to Congo and Sierra Leone. Is in the write-up.
Yes, sir.
How did you source the plant?
Yes, sir.
How did you source the plant for the experiment? Is it now domiciled in Nigeria or what? How did you source?
Yeah, we were able to get it here in Nigeria.
Where is the location? Which area? Which state?
In Enugu State.
Okay.
In Nsukka. Yes, sir.
Okay. So, there is no other question for you from here. Do we have anybody online that want to ask her a question? Anybody online?
Yes, sir.
Okay.
Except I have a comment, sir.
Okay, please go ahead.
Yes, as regards the figure that the comment was passed on, I think for the purpose of this presentation, figures are better. But what she has done wrong is not to put good legend on the figure. Because if she had put maybe the statistical significance on the figure to show that, because when you look at the figure, you can see at 30 minutes, you can see the effect of the drugs on inflammation. At 1 hour, you can see the effect. Are there a difference between the extract at 100 milligram per kg, body weight of the animal, 200 milligram per, because when if she puts table, you may not be able to really grasp what she's trying to say. I think I prefer this figure because of this presentation. But she has not been able to put what is needed on the figure to make us understand better. Thank you, sir.
Thank you. Okay, all right. So you can step down now. Share yourself. Share.
Okay, sir.
Thank you for your presentation. Who else do we have online?
Ohika Dominic, sir.
Huh?
Ohika Dominic is online, sir.
Ohika Dominic.
Yes, sir.
Have you paid the necessary fees? We didn't hear you. Hello? Ohika.
Hello, sir. Abakar Show is online.
Please, you cannot.
Hello, sir.
You cannot share yourself until we tell you to do so, please.
Okay.
So we are addressing Ohika Dominic now.
Yes, sir.
Okay. Have you paid your? When did you pay and how much did you pay?
I'm an undergraduate, so I paid 5,000 for the conference, and I paid on the 20th of this month of July.
20th of July.
Yes, sir.
Okay. Share your slide.
Okay, sir.
Okay. Please be fast. Be fast. There are people waiting. If you are not ready, who else is ready?
The Abakar Show is ready.
Please, Abubakar, we have had you once. Let me see if we can have another person. And if we have time, we come back to you, please. Let's just.
Amdalat Lawal online, ma.
Lawal.
Amdalat.
Okay, let's.
Yes, sir.
What of your fees?
I've paid everything, sir.
What and what did you pay?
I paid for conference.
How much?
Full conference fee, 25,000 on 29th of June.
What of membership?
I renewed my membership with 7,500 the same day.
Okay. Share your file.
Yes. All right, sir.
Yeah, we are still waiting for you. Okay, Hamdalat. Okay. You have 5 minutes. Go ahead.
Good afternoon.
Afternoon.
Chairman.
Yes.
All of that protocol duty observed. Alhamdulillah, Lawa from Kwara State University, Malite. I want to present on the topic salicylic acid mitigates salinity stress effects on germination and biochemical response in groundnuts. Introduction. Groundnuts is an important legume with a range of applications, including oil production, direct human consumption as food, and as animal feed due to its mineral and vitamin content. Soil salinity in agricultural land refers to the accumulation of high level of soluble salts within the root zone. This creates osmotic pressure, which prevents plant roots from absorbing water and restricts plant growth. It also interferes with the proper absorption of vital nutrients, which hinders plant growth and development. Salinization reduces arable land and threatens global food security. Therefore, utilizing marginal land for food production is essential as cultivable lands shrink due to urbanization, but soil salinity poses a major problem. So, eliminating excess salts from the root zone is necessary to improve crop growth, especially in salt-affected areas. And this could be achieved through various methods, chemical, biological, and physical methods. By a chemical method, one of the chemical methods is the use of plant hormone. So this work aims to evaluate the effect of salicylic acid on the germination of groundnut seeds under salinity stress, with a focus on biochemical stress indicator, including reduced glutathione, superoxide anion radical, hydrogen peroxide, and malodialdehyde on label. Materials and methods. The research was carried out at Physiological Laboratory, Department of Plant Biology, University of Illinois, and 2 groundnut varieties were examined, Samnot 11 and Samnot 14, obtained from Institute of Agricultural Research, Zaria. So there were 10 levels of treatment, 5 for sodium chloride, which include distilled water for control, 50, 100, 150, and 200 millimoles sodium chloride. And for salicylic acid, we have 1.5 millimoles salicylic acid only. Then salicylic acid for 50, 100, 150, and 200 millimoles sodium chloride. A total of 30 petri dishes for each variety, making a total of 60 petri dish. And each of the petri dish contains 10 sterilized seeds. And each of the petri dish received 10 ml of respective treatment solution. And these petri dishes were arranged in a complete randomized design, and they were monitored for 10 days. The parameters measured during the observation were the percentage germination, seedling length, which include radicle and plume length, seed figure index, and germination index. The biochemical stress markers determined were reduced glutathione, superoxide anion radical, hydrogen peroxide, and malodialdehyde content. So from the results, it was observed that the percentage germination increased with increase, and it peaked at 50 millimoles sodium chloride, which progressively decreases as the concentration of sodium chloride increases. And with the application of salicylic acid, the percentage germination significantly increased with the application of salicylic acid. Also, the plume length and radicle length also observe a significant effect of salicylic acid. In the sodium chloride treatment alone, the Samnot 11 did not record any plume length at higher concentration at 150, and even with addition of salicylic acid at 150 and 200, the plume length for Samnot 11 was 0. So this indicated that Samnot 11 is highly sensitive to soil stress compared to Samnot 14. In the radical observation, the increase in radical length and in both variety increases as the application of salicylic acid increases, and this was peaked at 50 millimoles salicylic acid plus sodium chloride in table 1B, in the seed figure index, it was observed that the seed figure index increased and progressively decreased as the sodium chloride concentration increases. And also, when the salicylic acid was added, the sodium chloride, the seed figure was significantly increased and also follow a reduction in magnitude, a reduction in the value as the concentration increased. The same observation was also observed for the germination index.
You have 1 minute.
As the concentration of sodium chloride decreased, the value also decreased. And for the salicylic acid plus sodium chloride, it actually increased the germination index, but as the concentration increases, for the sodium chloride, the value also decreases. Then
Go
for
to
the
your conclusion.
biochemical reaction, in conclusion, salicylic acid improved germination and seedling growth under salinity stress. It effectively mitigated salinity-induced oxidative damage, and antioxidant defense was enhanced. Out of all the parameters measured, the SAMNOT 14 proved to have a superior tolerance to salt stress compared to SAMNOT 11, and it is highly recommended for salt-affected areas. Thank you, sir.
Okay, thank you for your presentation. Can we have your title again? Scroll to your title. Yeah, title. Okay. Your title emphatically puts it positively that salicylic acid mitigates salinity stress. Salicylic acid mitigates salinity stress effect.
Yes, sir.
On germination and biochemical responses. That's to say that this compound slows down or controls stress in this plant. Did I get that right?
I can't hear you, sir.
Oh.
Come again, sir.
Okay. Do we have any other question? I don't know how.
Hello, sir.
Hello. Hello.
Yes, sir. Yes, sir.
Your conclusion, you need to put it just like you said it. SAMNOT 14 exhibited better salinity tolerance compared to SAMNOT 11. Hello,
Sorry.
I hope you are hearing.
Sir. Yeah, yes, sir. I heard you, sir.
You need to type it inside. Right. Then what is this vigor index or vigor? What is it about? Vigor index or vigor. Can you explain what does it have to do with? How did you work on it? How did you calculate it, or what does it imply?
The seed vigor index was calculated by using percentage germination and seedling length. The percentage germination we calculated, then we multiply by the seedling length. The seedling length we consider here, we consider the average of the both plumon and radicle added together and multiplied by percentage germination to get the seed figure. The seed figure index.
What physiological status does that signify? The vigor.
It signifies the performance of the seed.
Okay. Okay.
It signifies the performance of the seed. So high seedling figures indicate vigorous and stress tolerance of seed.
Okay. Okay. Do we have anybody online that has a question or comment for her? Yes. Anyone listening? Okay. Uh, uh, Lawa, thank you for your presentation. Thank you. Thank you. So you can unmute yourself now. I mean, unshare yourself. Unshare. Okay. Who else do we have ready online?
Hello, good afternoon, sir.
Afternoon.
This is Jafar Khadijat Suleiman.
Okay. Have you paid your necessary fees? Have you paid your fees? Have you paid the conference fee, membership fees? Khadijat, are you hearing us? Unmute yourself. Unmute yourself. You have muted yourself. Unmute.
Okay, I can hear you, sir.
Okay. Have you paid the necessary conference and membership?
Yes, sir.
How much did you pay?
15,000 naira.
Okay.
As for graduates.
What of membership?
Yes, together with the membership, that's 15,000.
Oh,
10,000 early bird for postgraduates.
okay, okay. Which account and date?
Date, let me check the date.
Okay, it's all right. Which account?
One Echo Bank and the other one is another bank for the membership.
Okay, beautiful. It's all right. Share your file and then you have 5 minutes.
Okay, sir.
Yeah.
B.
We are not seeing your file yet.
I want to share it now, sir. Sorry.
Okay. Jafar.
Sir, I'm still looking for the file.
Okay, can we step aside and come back later?
Okay, okay, sir.
Okay. Who
Okay,
else is
thank you.
ready?
I'm ready, sir.
Okay, who is speaking?
It's Ugume Soma.
Okay, Kunle.
Ugume Soma.
Okay, Mesoma. I think I get
Yes,
that
sir.
clearly. Have you paid the dues and the conference?
Yes. So I'm an undergraduate. I just paid the 5K for the registration.
Okay, okay. Okay, okay. All right. Share your file.
Okay, sir.
Is that Jafar?
Can you say it, sir?
We're seeing something. We don't know who we are seeing. Put it on presentation mode. Presentation mode.
Okay, sir.
It's too tiny. That's why we can't see. Who do we have there? Can you see it? We can't see it like this. Put it on presentation mode.
Okay, sir, that's what I'm trying to do.
Okay. Who else is ready? Who else? Who do we
Dominic,
have?
Ohika Dominic, sir.
Who?
Ohika Dominic, sir.
Ohika Dominic. Okay.
Yes, sir.
Have you paid the necessary fees? Answer the question.
Yes, sir, I've paid. I've paid, sir.
What did you pay? How much and when?
I paid 5,000 on the 20th of July.
Okay, share your file.
Hello, sir.
Share your file, Ohika.
Okay, sir. Okay, sir.
Okay, Ohika, you are not ready. You are not ready. Who else is ready? Jafar, are you ready? Jafar? Who else is ready?
Agnes Uyoyo. I'm ready, sir.
Who? Who is
Uyoyo Agnes. Aporobaro.
Agnes. Okay.
Yes, sir. Dr. Uyoyo Agnes Aporobaro.
Okay. What did you pay? And when?
I paid 20. I paid everything to the BOSON account, 32,000.
Okay. As what? What's the breakdown?
I paid 20.
Okay.
For no, the breakdown, what I did there, though the Professor Akama said I made mistake, but say we correct it. I just paid everything, 32,000 into the BOSON account. 32,000, everything into the account. Both the actual 2,000, the 10,000 for the conference, and the registration fee, 20,000 naira. I paid everything there. And it told me,
when you say BOSON account, in which bank?
It is which bank are they? I'm coming. You know UB, Abi. Yes. I don't know, but there are two accounts. There is one Echo. There is one either Unity Bank. I can't remember, but I know I sent it to Professor Akoma. Akama, my receipts.
Share your file. Share your file.
I'm coming, let me share it. This is so far. Dishes.
We're waiting.
Okay, sir. Huh?
Please be fast. People are waiting.
Okay. All right.
Agnes, you are taking time.
Please, I'm coming. No, just sit down. So far. Yes.
Agnes.
The thing is slow now. It's like I don't like network or something. Just wait.
Okay, it appears it's loading now, but we have not seen
Yeah.
it. Oh.
Loading. Yeah.
We have not seen it.
Stop.
It's loading. It's loading, but we have not seen it.
Okay. This one straight stop fine. Stop fine.
As it is, it's like we will have to come back to you.
Okay, sir.
Yeah. So unshare yourself, unshare.
Okay.
Unshare yourself. Unshare, unshare. You are still featuring. Unshare, unshare. We'll come back to you. We'll come back. People are waiting. People are waiting.
Okay, sir.
You need to clear from the screen.
Hold on, sir. I want to.
You are imposing yourself. You are imposing yourself. Studio, please take control. Studio, please take control. Take control. Thank you. Who do we have that is ready?
Azumi Obama.
Who? Dan Azumi?
Dan Azumi Obama.
Okay. How much did you pay and into which account?
I paid 2,000 for abstract submission.
Good.
10,000 for membership.
Good.
And 10,000 for BOSON outside university.
Okay, share your file.
Yes, sir.
Yes.
Can you see it now, sir?
We are not seeing it yet. We are not seeing anything. Share your file. We are not seeing anything. Yeah, I know many of you out there are students. You are just trying to get a handle on these facilities. What you do is you create an event, you and your friends, and learn how to navigate these things just among yourselves. It's not when you come out here, you are facing the world.
Can you see it now, sir?
You'll be fiddling. We cannot wait.
Can you see it now, sir?
We are not seeing you. We are not seeing you. So please, who else do we have?
Huh?
Who else do we have that is ready? Okay, let me go back to Dr. Doc. Is it Kazauri? Karai. Dr. Karai.
It's Abakar Show.
Yes.
Hello, sir.
Hello, sir.
Can you hear me?
Dr. Karai.
Please, is Mala Bakshi.
Go ahead. Go ahead. You have 5 minutes. Go ahead.
Okay, sir.
Yes.
Okay. So all protocols duly observed. Okay. Okay. Hello. The topic I'm going to present is comparative nutritional and mineral profile of the seeds of Strilus lanatus and Cucurbita maxima grown in Sokoto.
Wait.
Introduction.
Wait. You have your phone there with you, tuned online, and you are causing echo.
Okay.
Switch off any other
Okay, sir.
gadget that is tuned to this line.
Okay, I have done that, sir.
Okay, go continue.
So, introduction. Malnutrition remains a major public health concern in Nigeria, particularly in northern regions such as Sokoto, where protein and micronutrient deficiencies are prevalent. Seeds of indigenous crops offer a sustainable source of nutrients that can supplement diets. Among such crops is Citrullus nanatus, watermelon, and the Cucurbita maxima or squash are widely consumed and cultivated in the region. Both seeds are byproducts of widely consumed fruits that are often discarded despite their nutritional richness. Previous studies have reported their richness in oils, protein, minerals, and bioactive compounds. This study aims to evaluate and compare the proximate and mineral composition of C. lanatus and C. maxima seeds grown under the agroecological conditions of Sokoto.
Studio,
materials
please.
and methods. Sample collection
Studio, please. Take control, studio. Take control, please. File him off.
Materials and methods.
He cannot obey simple instructions. He can't obey simple instructions. Please remove him. Yes. Who do we have that is ready? Dan Azumi. Dan Azumi, are you there?
Yes, sir.
Okay. Show yourself. And have you paid the necessary dues?
Yes, sir. I've done that, sir.
What did you pay and how much?
I paid 2,000 after submission,
Okay.
10,000 to Benson Edausa University account,
Okay.
10,000 for membership registration.
Okay. Share your file. Share your file. Share your file. Yes, Dan Azumi.
Yes, sir.
Yeah, go ahead. Go ahead.
Okay.
You have 5 minutes.
Okay.
Wow.
Biodiversity and phytochemical analysis of medicinal mushroom species of Niger South Senatorial District, Nigeria. Mushrooms are microfungi with distinctive fruiting bodies, which can be hypogynous or epigynous. Mushrooms contribute to ecosystem dynamics, human nutrition, and traditional medicine. Scientific interest prompting investigation into the intricate interplay between mushroom biodiversity and their phytochemical composition. Mushrooms are spore-bearing fruit body normally formed above the ground. They have deep-rooted history in Nigerian traditional medicine and culinary practices. Mushrooms are recognized globally for their medicinal and nutritional properties. There's limited scientific documentation on the types, distribution, and phytochemical composition of the mushrooms in part of Niger South Senatorial District. Mushrooms possess a challenge to their conservation and proper utilization. Indigenous knowledge is at risk of being lost if not properly documented. Their importance will not be harnessed, and for that they face a lot of challenges. This will contribute to biodiversity conservation by documenting underrepresented fungi species in Niger South. It will support the search for a new bioactive compound. It will also help in preserving indigenous knowledge of medicinal mushrooms, and it will stimulate economic development through the cultivation and commercialization of valuable mushrooms. The aim of this research is to study the biodiversity and phytochemical analysis of medicinal mushroom species in Niger South. And the specific objective is to identify and document medicinal mushroom species, classify the identified mushroom based on morphological characteristics, conduct phytochemical analysis of selected medicinal mushroom species, and assess the potential medicinal relevance of the identified phytochemicals. Study area. 3 emirates in Niger South were selected: Bida, Agay, and Lapai. Fresh mushroom samples were collected: Ganoderma aplanatum, Ganoderma tugai, Ganoderma lucidium, oyster mushrooms, Trimeted versicolor, Daldinia concentrica, Podasis pistellaria, Lycopodium periform, Ganoderma sicile, and Letinuna edod were collected from the wild in different locations in Niger South. Materials such as knife, hand gloves, weighing balance, tissue paper, sample bottles with 10% diluted formaldehyde were used for their preservation. methodology. Clean and dry under sun. After the drying, they were grounded into powder and divided into two, one for proximate analysis and second for the phytochemical analysis. For the proximate analysis, crude fiber, ash content, protein content, carbohydrate content, fat content, moisture content, energy content were also identified using standard analysis procedure. For phytochemical analysis, tannin, flavonoid, alkaloid, saponin, terpenoid, steroid, glycoside, phenol, anthraquinone were also identified using standard analysis procedure. Result and discussion.
You have 1
Protein
minute.
content, that is,
You have
you
1
said?
minute. 1 minute.
Okay. Protein content Latina headed had the highest value with 29.73 and Ganoderma tussai with the lowest 6.21%. Fiber content were also identified for oyster species. Carbohydrate content of Ganoderma tussai still the highest with 70.96% and the Ecopedium periform the lowest. In fat content, Primet versicolor had the highest value content, 4.693%, and Ganoderma lucidium the lowest. Moisture content range from 3.09% for Ganoderma lucidium to
Go to conclusion.
8.8.
Please conclude.
In conclusion, the overall linear concentrica, Lycopodium periform, and Leutinella headod stood out with the highest phytochemical concentration in different categories, while Ganoderma aplandum showed the lowest level across most phytochemicals. This variation reflects the diverse bioactive profile of the mushroom species. Thank you, sir.
Okay, thank you. Thank you for your interesting study and presentation. Okay. Can we go to your title page?
Okay.
Yes. Okay. This biodiversity and phytochemical analysis of medicinal mushroom species, Niger South Senatorial District of Nigeria. Let me first of all point out analysis as spelled here will have E instead of I.
Okay.
You get that? Okay, because it is plurals. It's not just one analysis you are doing. You are doing more than one analysis. That's why.
Okay.
Go to your methodology slide. I think it's the third one.
This very
no next. Go to your methodology slide. You are so slow. Keep going. Keep going. Yes. Okay. Okay, this is showing the two different treatments that you are applying to the mushrooms. Is that not so?
No.
The treatments you are applying to the mushrooms.
Yes, sir.
Okay. All right. Go to the next. Next slide.
Okay.
Okay. On this your result, the first bullet there. Why did you credit it to Bilal and Oyetayo?
Yes, they also, it's in line with what they also have from their literature, sir.
Is that why you put it like this?
Yes, sir.
No, you cannot do that. Because
Okay.
what this is saying is that that bullet information, that information in that bullet is from these people. It's not your work. It's not your discovery. It's not your finding.
Okay.
Do you get it?
Yes, sir.
You can refer to them and mention that they have acquired similar results in your discussion, but not when you are presenting results like this. Is that clear?
Not yet, sir.
Okay, now Zumi.
Yes, sir.
The species you work with, how did you identify them?
I use mushroom guide. And mushroom guide.
Okay, mushroom guide.
Yes, sir.
Okay. So it is based on the guide that you have you arrive at this. Who authenticated this species? Because I believe
I use mushroom guide and also
they are land races probably collected from the wild.
Yes, sir.
And if using the guide,
mushroom application, also sir, mushroom identification application.
Okay.
Yes,
so you arrive at this.
Yes, sir.
Who guided this identification, or based on the tools you use, you arrive at these names? Because identification, especially for mushroom, I know is something which is not straightforward like other plant species. So you need to be sure of those names and species that you have. Otherwise, there will be a problem. The edible ones that are known, or these oysters that are there, that one cannot be contested. They are well known and identified. But collecting mushroom from the wild and arrive at this identification, that's where I raise. So please, you need to look at that so that you present something which people can rely upon. Thank you.
Thank you very much.
Hello.
Hello,
You said they are useful in traditional medicine. I hope you are hearing me.
I'm hearing you, sir.
You never mentioned the diseases that they are used to treat. And you said they are used for their culinary properties. I would have loved maybe you go further and say something like in soup, stews, and sauces. And this particular fiber content area, though before I get to that word, when you are listing your ganodermas, the moment you are listing up to 1, 2, 3, 4 ganodermas, the next one will just be capital G full stop. You don't keep on repeating ganoderma, 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 ganoderma.
Okay, sir.
Also, there's a place there where there is a continuum where you mention up to 3 ganodermas at the same time. Then on this place, let me just finish. Where you have this fiber content, you wrote oyster species. What I do here is oyster mushrooms. I've never had direct oyster species before. You have to check on that.
Okay.
You found many Ganoderma species. Which one of them is most popular or important to your knowledge? Okay, thank you, Dan Azumi. Thank you, thank you, thank you. You can unshare yourself, unshare yourself.
Okay, thank you.
Unshare, unshare. Please, studio, take control. Okay, who do we have next? That is.
Good afternoon, sir. I'm ready, sir.
Who am I talking to?
Miss Soma.
Miss Soma.
Miss Soma.
Okay.
Yes, sir.
So, did you pay the necessary dues?
Yes, I paid 5,000 naira. I'm an undergraduate.
Okay, share your file.
Okay, sir.
Yes, put it on presentation mode.
Okay, sir.
It's still too tiny. It's still too tiny. Landscape it.
Okay, sir.
When we left you alone, we thought you will take the time to master what you need to do.
I'm on it, sir.
Okay, it's better now. It's better. You can continue from here.
Okay, sir.
You have 5 minutes.
Okay, sir. So my topic is the phytochemical analysis and antioxidant properties of the methanol extract of the roots of Combretum murrayanum. And Combretum murrayanum is a strangling shrub native to Sierra Leone and Congo, and the common name is bush willow. It belongs to the family of Combretaceae. And the medicinal use of this plant is of high interest in the African continent. Like the aim of the experiment was to assess the qualitative and quantitative phytochemical constituents to determine the LD50, to assess the in vitro antioxidant potential of the crude extract, to assess the in vivo antioxidant potential. The materials and method. The plant collection, the plant was collected, it was extracted, and then used for the analysis, for both for the in vitro and the in vivo parts. And this is the result for the qualitative phytochemical analysis. All these phytochemicals were found present in the plants. And this is the quantitative part. Then this is the in vivo results showing the MDA, the sword and cat method. And then this is the results for because it was done for 3 weeks. So this is the results for week 1, week 2, and week 3. And this is the table for the in vitro parts using scavenging acid. This is the chart, and this is the result for reducing power, nitric oxide. Then discussion, it was actually carried out to assess the phytochemical analysis and antioxidant properties of Convertum murianum using methanol for the extraction. And the qualitative phytochemical analysis showed presence of many phytochemicals. So it suggests that like Convertum murianum, it may not be as effective as ascorbic acid in terms of an antioxidant property, but it still shows that it possesses some traces of antioxidant properties. Thank you.
Okay. Thank you for your
Yes, sir.
presentation. And thank you for keeping to time.
Thank you.
But I did not see the results because you literally throw it at me.
Okay, so let me go back.
It's not about going back.
Okay, sir.
It's the mood of the presentation. When you show a table, you need to tell your audience what the table says. You cannot expect your audience to interpret the table. You are the one to interpret it for them. Like this converging activity graph you are showing. What does it mean? Can you explain this?
This is just like showing the graph for both the extract and the ascorbic acid.
Yes. So what's happening to the extract? What's happening to the ascorbic acid?
It shows that like the ascorbic acid is like it has more antioxidant properties than the extract.
Okay. By how much?
No, it was not stated here.
So, why will you not state it? You are just you are coming up, you are learning, but I'm trying to tell you that it takes more. You need to do more than what you have done, just throwing the data at your audience. So, for example, this graph you are showing,
yes, sir,
is showing the profile of the activity of the extracts and ascorbic acid at different concentrations and their percentage inhibition. So the graph shows that ascorbic acid activity profile was higher than that of the extracts, but they have a similar profile with different concentrations. Do you get that?
Yes, sir.
You see? So you need to tell people the information in the data before you move on, so that they can be following you. Are you clear now?
Okay, sir.
Okay.
Yes, sir.
Okay, thank you for the presentation. apart
Yes,
from
sir.
the ascorbic acid standard you use, which
Yes, sir.
other one, other assay for evaluating antioxidant activity that you think you can use?
Is it okay apart from ascorbic acid? I think like fruits,
Eh?
like fruits,
Like what?
fruits.
Like fruits. That's
Yeah.
not what I mean. In your comparative evaluation of the plant extract, you use ascorbic acids. There are other assays for evaluating antioxidant activity. Why did you resolve to use only ascorbic acids as a standard?
Okay.
In the,
Ascorbic acid was actually what was available
no,
to us to use.
in the literature search, what are the other available methods that are used to evaluate the antioxidant potential of plant materials? Did you come across any?
No, sir.
Have you heard of BPPH, radical scavenging?
Yes, sir.
Did you heard of PRAF assay?
No, sir.
Okay, so there are many things. So at times you use only this. Comparatively, you see the ascorbic acid are far from better than your extracts. You could have
Yes,
try another method so that you compare that, okay, using this assay, this is the result that you obtain. And your graph did not in any way clearly spelled out the percentage inhibition so that I will see whether that is significant in any way, because this is a crude material. When you use crude material, even at the same concentration, you should not expect that to give you the same type of potency. So
Okay.
that's where you diversify methods and get to know at what concentration. You should not expect to have ascorbic acid at 4,000 or 5,000 microgram per ml. You compare it with the extracts and you say your extra is non-performing. Remember, ascorbic acid is already fully refined chemical, which is known to have that. So
Yes, sir.
don't underrate your plant by comparing it
Okay.
with this. Thank you.
Okay, sir. Thank you, sir.
You said this plant is a strangling shrub. Yeah, you never show the pictorial or the imagery. Is it a recently introduced combrentum plant into Nigeria?
Yes, it's a novel plant, sir.
A novel plant
Yes, sir.
that is recently introduced.
Yes, sir.
You should have buttressed with pictorials, imagery of the combentrum. More so when you said it's a strangling shrub, so that we can see what you actually intend.
Okay, sir.
So, thank you. Thank you for your interesting presentation. You can stand down now, unshare yourself.
Okay, thank you, sir.
Okay. Who is ready?
I'm ready, sir. Patience, Olayinka Beniwabo is ready, sir.
Patience.
Olayinka Beniwabo, I'm ready, sir.
Okay, okay. So, have you paid the necessary fees?
I did.
How much? The treasurer is here to hear your record. How much did you pay and to which account?
I paid 20,000 for BOSON virtual presentation to, I think, Echo Bank, and Professor Chris or something is aware of it. And I just paid 7,500 this morning. And Dr. Kolawole is also aware of it.
Okay, okay. The name again?
Sir?
Your name again?
Patience Olayinka Benuwabo from Kwara State University.
Okay. Share your file.
All right, sir. I want to share my file.
We're waiting for you. We're waiting for you. Share your file.
I'm trying to share.
While we wait for her, to all our students, yeah, you will start one day or the other to learn about these things. But one easy way to do it is to plan it with your friends. Then together you'll be learning how to share, how to connect. You know, you create an event because it's possible. You create an event for yourselves. It's not when you come to an international conference,
I'm on, sir.
we will now be waiting for you. There is no time to do that. So we're just tolerating this for now. You need to have schooled yourself on all these basic things before getting to this point.
I have a question.
Go ahead. Yes.
I have a question.
Yes.
Hello?
Ask your question.
Please, I am Dr. Marie. I was supposed to present yesterday, and I was number 17 on the list. I've been tabling my question, my problem on the group chat, and nobody seemed to answer me. I've not yet presented. I was supposed to present
Are
yesterday.
you the one we just gave space to present? What's your name? What is your name?
Dr. Nyanju Yomi Marie.
Okay, okay. Are you ready right now?
I am ready, sir.
Go ahead.
Sure.
Did you make the necessary payments?
I made a necessary payment. I even showed my DC on the DC. Dr. Luchi, on my on the WhatsApp, I've shared everything. I've submitted everything.
Who is coming now?
Oh? Yes, I want to share my slide now.
Go ahead, go ahead. Okay, Patience. We have Patience.
Yes,
Olayinka.
Yes, sir.
Okay, go ahead. You have 5 minutes.
All right, sir. I'm presenting my paper on spatial distribution and ecological risk assessment of cadmium, copper, and lead in agricultural soils of Ilori, Nigeria. And I'm from Department of Plant and Environmental Biology, Kwara State University, Malete. My outline goes thus: from introduction to aim and objectives, materials and methods, results, discussion, conclusion, recommendation, and references. So my introduction now. Heavy metal contamination of agricultural soils we know poses significant threats to food security, environmental quality, and health of humans. And due to urbanization, sounds like Ilorin in Nigeria's agricultural land are posed with pollutants from various anthropogenic activities, like industrial activities, vehicular emission, and the rest. Now, this study investigates the spatial distribution and ecological risk of those metals mentioned, copper, cadmium, and lead, across some major agricultural lands in Ilorin, Kwara State. Now, we analyze metal concentration and apply some ecological risk indices, actually to study the assessment of the contamination level of this aforementioned heavy metals in order to be able to identify pollution hotspots in those farmlands and to also provide detailed information for sustainable land use and environmental protection. Now, what are my aims and objectives? I want to do this. Excuse me. Now, my aim and objective is to assess the distribution of these metals in agricultural soils, to be able to evaluate via soil indicators the possibility of this land to provide quality food for Ilorin dwellers, so as to guard against food scarcity and land use, to also be able to provide constructive land management for agricultural purpose, which will inform availability of food for the citizens of Ilorin, Kwara State, and the environment. Now, my material and methods. My material and methods goes thus. I sampled some soil from these major agricultural soils, and I follow standard protocols. And samples were dried, saved, and subjected to physical-chemical properties analysis and heavy metal screening. And these are my models. My model used to quantify and evaluate my potential ecological impacts of these metals on this soil sampled are as below. The contamination factor of these soils are also assessed, and the ecological risk indices of these soils were also estimated by this model. This is the interpretation. Now, should the cumulative risk index is greater than 150, it is tend to show a low risk of such heavy metal. And if it is over greater or less than 300, is said to be at moderate risk. And so also do we have a considerate risk and very high risk indices for these metals.
You have 1 minute. 1 minute.
All right, so these are my results. My results were analyzed by bar charts, principal components analysis, and the box plots index, and then cluster dendrogram to show the spatial distribution of these key physicochemical properties as a function of variability in different sites where the soils are sampled, to be able to show the status of the agricultural soils in Ilorin metropolis in order to inform the kind of produce from those soils, and to also support agricultural soil management and strategy and results.
Go to your conclusions, please.
All right, sir. My conclusion. These studies offer a detailed evaluation of cadmium, copper, and lead contamination in agricultural soils in Ilorin, Nigeria. And this results of a study also uncover pronounced spatial variability in both soils, physical attributes, and associated ecological risks. Integrating multi-status analysis enabled me to identify critical contamination hotspots from these sampled soils. And it also showed the fact that the soil chemistry governs the metal mobility, which informs how contaminated these soils are in indices to producing quality products based with these soils, and also environmental sustainability. The principal component factor also allows me to see the cluster profile of these metals in these soils sampled. Together, these findings point to urgent need for tailored soil management strategies, consistent environmental monitoring, and adoption of sustainable farm practices. Such efforts are essential to reduce heavy metal contamination and also to safeguard both health of the agro-system and the well-being of the local community that depend on the food produced from these soils.
Thank you.
Thank you.
Thank you very much for your interesting presentation. Though I have to be a little bit brash with you because I wasn't expecting this. You did not manage your time well, and we expected for your caliber to do better than this. Please, next time. You know
right,
now, KYA, know your audience. On a panel where I have more than 7 professors listening to you, what are you doing with introduction methodology and neglecting your results? You should know now. This is what we are expecting your caliber to be passing down. The awareness of the audience. So, having said that, your presentation was good and interesting. I just pray you'll do better next time. Please, we respect you and the work you have done. Okay?
My question is, what is the implication of the dendrogram that you got from your cluster analysis? Are you there, Patience? Hello, Patience. Hello? She's not there, clearly.
Hello, Patience, are you still there? Hello? Hello? Okay, studio. Take control.
I'm there talking, sir.
Okay. We were not hearing you. Are you still there?
I'm there. Yes, sir, I'm there talking.
Okay, we are not
Yes,
hearing
answering
you.
the question of the dendrogram. Answering the question of the dendrogram. The dendrogram is able to indicate.
Okay, now we are not hearing you again. Patience. So,
the fact that there is site-to-site specific contamination of this method, that anthropogenic activities differ from. So,
all right. Thank you very much, Doc. Thank you very much.
Thank you, sir.
Yeah. Okay. Who is next? Unmute yourself and.
Okay. I am ready, sir.
Thank you, sir.
I'm ready, sir.
Thank you very much.
Okay.
I'm ready, Dr. Agnes.
Who am I talking to?
Dr. Agnes. Uyoyo.
Who?
Uyoyo, Agnes. Dr. Uyoyo, Agnes.
Okay, Agnes, you are ready now.
Yes, sir.
Okay. Share your file.
Okay. Are you saying it?
No, not yet.
Okay. All right.
We are not. Okay, it's coming. It's coming.
Sir.
Not. It's coming.
Are you saying it?
Not yet, but it's coming.
Okay.
It's coming.
All right.
We have not seen it, but it seems to be coming. Click on the screen. Click on the screen.
Okay.
It's not showing, no.
I'm coming.
I thought you would have taken that time to get to
No,
one
that.
side and master this before you come back. We have people waiting online, so we cannot wait for you. We cannot wait for you.
Maybe it's network.
Network is there.
It's network problem.
The network for you, what's it going over? People there.
It's network problem.
It's a problem.
We are not seeing your file,
We are coming.
That's what you do as Nigerians. You will be
Can you
going
see it
there.
now?
Okay, we can see you now. Yes, we can see you. Just continue like that. You have 5 minutes.
All right, sir. Okay. The title of my seminar is Eco-Friendly Enhancing the is eco-friendly fertilization enhances soft fertilization, fertility, and growth of Salonum zephyrson using soil human urine under rainy season cultivation in Agbo, Nigeria. Introduction. Soil fertility is a crucial factor that influence agricultural. Materials and methods. So the material used were collected from cultivated lands. Then the urine were prepared, was urinated by or peeled by the corresponding author, and it was stored for 17 days. It was applied to soil. The treatment for the soil, and we have the 360.1 mil. We also have 620.1 mil of urine. Then the control, no treatment. So they will apply to this soil. After applying this, the water leaf teteria, which is known as seed, 2 seeds were planted in each pot. After germination, one will remove, remaining one. Then the results of the physiochemical parameter of the soil indicates that this urine improve the soil fertility, which reveal in the plant growth. So we can see on the tree here, the stream here, the results. The control is the column 1. The test soil analysis before application of urine is the column 1, while the control is the column 2. The 360.1 mil is the column 3, while the 620.1 mil is the column 12. So looking at this chemical parameter analysis for the lab, we can see that the 620.1 mil of urine soil improve pH, which is 8.1. You can see the sulfur, they increase the macronutrients. The sulfur there is 5.57. The phosphorus is 8. The nitrogen, which helps to improve plant growth and yield, is also 8. So, but compared to the 360 mil soil, the phosphorus there is 7, the nitrogen there is 7. Then what about the control? The control soil is 0.01. Then the soil tested before application of urine is the nitrogen there is 0.03. So looking at this result, we can conclude that the 620 mil of urine improve this soil fertility or the micronutrients in the soil. Then the result, the morphology of the plant so in this soil. The first place that you are saying is the control place without urine treatment. Then the second one is the 360.1 mil of urine in the soil. Then the third plant you are saying there is 620.1 of urine in the soil. Then looking at it, you can observe that the physiochemical parameter in the soil reveal is evidence in these plants. The plants there are 620.1 mil. You see the way they are robust, greenish, fine, solar, compared to 360, totally as scanty. But when we talk about the teteras, that is the seed of water leaf, which is the seed. The 360 mil of urine improve the yield. While the vegetative growth, the 620 mil of urine improve the yield of the 620 growth. Discussion. There are many researchers that have worked on this, although many of them use raw urine, but the one they used was stored human urine. Why did they store human urine? After, when the urine contains any disease from the source, the person that provides it, during storage fermentation, it removes all this sickness from it, or removes this from it. That was the reason I stored. And I can also observe that from most online work, they use raw urine and say hi.
You have 30 seconds. 30 seconds.
Okay. Conclusion. The methodology employed in this study, which involved the application of 2 different volumes of 70-day stored human urine, addressed the issue faced by agro agriculture in Nigeria. The results showed that the application of 360.1 mil and 620.1 mil of stored human urine improves soil physiochemical properties such as the pH, the nitrogen, the phosphorus, and others. So the conclusion is that agricultural or local farmers they are advised to use stored human urine as fertilizer alternative to improve plant growth. Like fertilizer, the synthetic fertilizer, they degrade this our soil, but this organic manure they help to improve this soil and also help to give the plants that it will produce added nutrients.
Thank you
Thank you for
for
listening.
your very interesting presentation. Thank you so much. Do I have anybody that wants to ask?
Thank you very much. I hope you are still hearing us.
Yes, sir. I'm here, sir.
I want to believe that your Talinum fructosum is likely going to, is likely going to be the same thing as Talinum triangulare.
Triangular, yes. Triangular lane.
You should have put synonym. That is what we call synonym. That is means the same thing. SYNONYM. Then I believe that you should have also put the family that it belongs at the vegetable level. Then you said the terraria. And you answered it as the seeds.
But the fruits of the
maybe the seeds or the fruits. But it's not universally acknowledged as terraria. Then your number 17, there we have an index something where you wrote capital C, capital E, capital C. What does that one mean? CEC. Where you are listing your nutrients. Boron, copper. Then the last one, I will suggest that you take the experiment further to the level of a concentration. You said one is 360, one is 620. Why don't you take the experiment further to the level of 1,000 mils of urine and see whether there will be depreciative results? I hope you are getting what I'm saying.
Yes, sir.
You are trying to say that with the 620, you have the best result. Then with the 360, you have mild result. Can you afford...
But it's mild result with high fruits, with high terera.
Then you should, I would have wished that you should try 1,000 ml to see whether there will be more increment or there will be degradational results. I hope you can get me.
Yes, sir.
Then what do you mean by that 17? Capital C, capital E, capital C. Thank you.
That's our your exchange capacity. Can you hear me, sir? Cation exchange capacity.
We got you. Thank you.
All right, sir.
You actually suggested that this treatment can substitute for inorganic
fertilizer. Yes, sir.
How feasible is that?
Because look at the nitrogen. When we look at the nitrogen here, the parameter from nitrogen, the value, the nitrogen is high.
I mean, how feasible? How much can you get?
How much urine?
Yeah. Yes.
Okay, the 360 urine improve the vegetative growth. So if we are substituting this for inorganic fertilizer or as alternative used, then we should use 360 of stored human urine 17 days. 360 for vegetative growth. Then 620 360 for reproductive growth. Why 620 for vegetative growth of this stored human urine?
Okay, we have a question here.
You are still listening?
Yes, sir.
Have you ever considered the psychological effect of the buyers knowing that these are urine-grown water leaf? What can you say about that?
Like before I carried out this experiment, I went online and there are reports on using raw urine. I read on raw urine your raw urine, it has some biological effects, especially when the the source you get it from is is suffering from a particular illness. So when you apply it to soil, it's made, but the plant depends on soil. The plant can absorb it and it can become biotransformation. So people that will consume it. Then the one fermented, during fermentation, not less than 10 days. Like it should be from 10 days to 1 month, no more than 1 month, unless it lost the nutrient. That this is that refine this urine and give it a good, give a beneficial nutrient that will help plants. That will help plant when you plant, the plant can absorb it. Now the plant will improve plant growth, not only improve plant growth, and also it will also improve the nutritional value for the plant. How do we know? People, those some researchers that use 200 ml of urine, they retard growth of that plant. So, but this is not retard growth. Why?
Okay. I see your determination and enthusiasm on this subject. We're just trying to draw your attention to the difficulty of acceptance. So maybe you will learn to use the term treated to try to encourage more and more understanding of the subject and the approach when you are propagating the idea, because you said it's not raw, it's not raw urine, it's
no.
So
Fermented
that
urine.
will be like treated somehow.
Treated.
So you will learn to use that term, to encourage people to be more amenable to the idea, rather than continuing and just mentioning urine. Your
Anyway, sir,
determination.
this one will be published. I will publish under the Nigerian Journal of Botany.
Okay.
So you can read more there about it. Yes, the everything
Yeah,
is there. The thickness
going forward, you know that your job as a scientist has not ended with just the output of the result. You have to convince people to accept it and use the idea. So till you start getting people to embrace the idea, you have not concluded your task. So we are just trying to guide on how the idea can be pushed easily to further acceptance. So thank you very much, Dr. Patience, for your...
It's not directly or something.
Sir?
Who is speaking?
Okay.
Yes.
So thank you, Doc. Unshare yourself. Unshare yourself. Okay. Unshare. Unshare. Unshare yourself.
Unshare yourself. I'm ready now. I'm ready now, sir.
Okay, go ahead. Go ahead. Share your slide.
All right. I'm sharing my slide now.
Okay.
Can you see the slide now?
No, not yet. We're not seeing it yet. Okay, it's coming. Good.
Yes.
We can see you. Go ahead. You have 5 minutes. Yeah.
I am Nyanju Yomi Marie Carol from Federal University. So my topic is on mobile phone as a vector of antibiotic-resistant bacteria. A case study of among the Federal University of BOSON students. So, we all know that mobile phones are quite indispensable personal items that frequently come into contact. That frequently come into contact, with the human body and various surfaces, and as such, it creates these opportunities for microbial contamination. So, because of the achievement, that's and a benefit of the, because we all know that mobile phone has so many achievements and the benefits. So it is easy to overlook. We always overlook it as hazards to health, which is against the background that many users may have no regard for personal hygiene, and the number of people who may use the same phone. So this constant handling of of the phone by different these users will now expose it to an array of microorganisms and make it a good reservoir of especially microorganisms that are associated, for instance, with the skin and with the skin. So, and also we all know that microbial resistance is currently has a great challenge worldwide. It decreases the effectiveness of drugs, that decreases morbidity and mortality, which is associated with serious and life-threatening infection, and thus compromising human health. So, what is actually the problem? The problem is that mobile phones, they are frequently handled and they are rarely disinfected and often carried into, environments such as toilets, kitchen, clinic, which makes them ideal reservoir for microbes. So, their constant use by students, as the case may be here, the case study of students in various settings like lecture hall, hostel, will now amplify the potential for microbial contamination. So, and despite the widespread of the mobile phone and their potential as vector for microbial contamination, we have realized that the scarcity of data on antibiotic resistant profile of the bacteria from the mobile phone. So, in a justification, we uh, we actually also realize that mobile phones are, there are so many in modern society, often serve as what? As an extension of the human hand, which give them, which give their frequent use and exposure to various environments. So the potential presence of these pathogens on mobile phone raise significant public health concerns. These bacteria can cause a range of infection, and the transmission of this pathogen through contaminated mobile phones could contribute to the spread of so many diseases. So, and also the multi-drug resistance is a common problem that hurdles chemotherapy. So, to overcome this problem, like of the multi-drug resistance, so we found it important to identify those multi-drug resistant organisms on the surfaces of mobile phones. So, here our methodology, the samples were collected. For the swabs, we use swabs. Swabs were collected from student handphones, and samples were prepared by enriching the incubate by enriching the swab and enrich the swab were collected and enriched by incubating them into sterile nutrient drops and incubated at 37 degrees Celsius for 24 hours. So the enriched samples
You have
were
1
then,
minute. 1 minute.
Oh, 1 minute.
Yes.
All right. Let me just show this is the table for the isolation, the occurrence of bacteria, how they occur. They were we got 12 isolates of E. coli, Staphylococcus aureus. We have up to 20. We have Klebsiella species. So these are their antibiotic resistant, the antibiotic resistant pattern. We actually saw that they were actually resistant to ampicillin, cefotaxime, and tetracycline. We all know that these antibiotics, they are these antibiotics, they are commonly used. They are commonly used antibiotics, and for them to be the organism to be actually resistant to those
Please
organisms that
go to your conclusion. Conclusion.
all right.
Conclusion, please.
Yes, I'm coming, sir. Sorry. There's something that the
Hello?
conclusion.
Please go to your conclusion.
I'm coming. Yes, conclusion. So the study had has a great evidence that mobile phones serve as a potential reservoir for multi-drug resistant bacteria, which include pathogenic of public health importance, like the E. coli, the Klebsiella, and the Staphylococcus, which are resistant to those antibiotics that I mentioned earlier. So we therefore recommend that that health education program should be instituted within the university to inform students about the hygiene practice, and regular disinfectant should be made on phone surfaces. And these are my references. Thank you for listening. Hello?
Yeah, thank you for your beautiful presentation.
All right.
Do I have a question? Please show us your title again. We were not able to get
Okay, I'm coming.
title.
I'm coming, sir.
You can voice it.
Okay. The title is Mobile Phone as Vector of Antibiotic Resistant Bacteria, a case study among university students of those
Okay. In one word. So, what actually did you find now? Is it a truism that mobile phones is vectoring antibiotic-resistant bacteria?
What we actually discovered is that we were able to isolate some microorganisms like this Staphylococcus or even this E. coli. E. coli is an organism that can be can cause urinary tract infection, and maybe we just realize that some people may be very careless about their phone. They carry it about. They go to toilet. You see some people even when they are going to toilet, they carry it, and when they go, they can displace it somewhere, and the phone will be contaminated.
What was your reference to euphobia poisoning?
Food poisoning?
Euphobia. Did you use any extract from euphobia?
No.
A plant called euphobia.
No, okay. I didn't want with plants.
Hello.
No, sir, I didn't want with plants. Maybe I could have included. It did not occur to me.
Maybe there is a mix-up somewhere. There is something about stem extract of euphobia poisoning reflecting in your. Okay, it's another person's work. Okay.
No, it's another person's work.
So thank you so much, Dr. Yanju, for your presentation. Thank you. You can unshare yourself.
All right.
Yeah. Good. Who do we? Khadija. I thought I saw her now. Khadija. Khadija, are you ready? Khadija Abdul Kadir. Are you ready? Abby? Oh, what's her name here? This name. That one is Yahuza. Khadija, are you ready? Khadija? Okay. Who is ready?
Abaka show.
Sir.
Abaka show. I have already started when you muted me.
You see, that time we who did we?
Hello, sir.
When we muted you that time, there was a lot of uncomfortable echo because somebody was watching the program online, either with a phone or laptop, right where you are.
I switch off my phone then.
You switch off your phone. Normal. There are other people around. We were the ones feeling the discomfort. You may not have been feeling it. So
yes.
if you have taken care of your background noise, then you can continue now.
Okay, sir. Thank you.
But once it repeats itself, please, because the noise was so very uncomfortable, the interference, you know.
I'm sorry. I'm sorry for that, sir.
Okay,
sorry for that.
Go ahead. Go ahead.
Okay, thank you.
Yeah. Go ahead. Go ahead. You have 5 minutes.
Okay. Okay, sir. All protocols duly observed. My name is Abubakar Shehu, Consultative Nutritional and Mineral Profile of the seeds of Citrullus lanatus and the Cucurbita maxima grown in Sokoto. Introduction. Malnutrition remains a major public health concern in Nigeria, particularly in northern regions such as Sokoto, where protein and micronutrients deficiency are a sustainable sources of nutrients that can supplement diets. Among such crops are Citrullus lanatus, that is watermelon, and Cucurbita maxima, that is commonly called squash, are widely consumed and cultivated in the region. Both seeds are byproducts of widely consumed fruits that are often discarded despite their nutritional richness. Various studies have reported their richness in oils, protein, milk by active compounds. This study aims to evaluate and compare the proximate mineral composition of C. lanatus and C. maxima seeds grown under these ecological conditions of Sokoto. Materials and methods. The sample collection mature fruits of C. lanatus and local. Sorry, something is. Hello, can you hear me, sir?
Assisting.
From local.
We are hearing you. We are hearing you. Just continue. Continue. We are hearing you.
Okay. Proximate composition analysis were standard method outlined by the Association of Official Analytical Chemists. Extraction method was used, and the ash content and carbohydrate was determined by difference. Mineral analysis, mineral contents were determined using atomic absorption spectrophotometry as after acid digestion with HNO3 and HCl4. It's okay. Excuse me, sir.
Yes, yes.
I think it's working.
What do you need?
My screen is working.
Okay.
Materials and methods, sir. The statistical analysis were analyzed using, the data was analyzed using SPSS version 2.6. Results were mean standard deviation. Difference between means were also a probability level of 5%. Then results in discussion. The table below shows that the parameters such as moisture content, ash, crude protein, crude lipid, available carbohydrate, crude fiber. In fact, all the parameters used show significance between the C Lenatus and C maxima, as the table indicated. And in table 2, it shows the mineral analysis of seeds of C Lenatus and C maxima, where the minerals such as sodium, potassium, magnesium, phosphorus, manganese, calcium, iron, copper, chromium, zinc, cyanide, nickel, all shows that there is significance and comparative analysis of C Lenatus and C maxima. Discussion. From the result, the moisture content showed range of values from 5.6 to 6.67%, higher than 4.78 to 5.21, and in pumpkin seeds, which was 5% as reported by. My screen is not showing.
Hello.
Obtained values were in agreement with 30.63.
Please conclude. Conclude, please.
Yeah. Okay, conclusion.
Your time is up.
This comparative study confirms that both Cereulus lanatus and Cucurbita maxima seeds are nutrient dense and possess promising mineral profile. Also, their regular inclusion in local diets and processing into flowers or oils as it shows significantly contribute to improving nutrition status and food security, especially in Sokoto State and similar agroecological regions worldwide. Also, it is recommended that public health initiatives should promote the utilization of these underutilized seeds for both human consumption and industrial application. Thank you.
Well done for the presentation. I hope you are hearing me.
I can hear you, sir.
In your write-up, you wrote, yes, this place, introduction. You said seeds of indigenous crops offer a sustainable source of nutrients that can, that can supplement diets.
Yes.
I want you to know, so that place to me needs adjustment. I want you to know that Cereulus lanatus, it was introduced and it has now naturalized.
Okay, yes, sir. Yes, of course.
So it's only the Cucurbita maxima that we say that is indigenous. So I would have loved that that place should have just been seeds of both naturalized and indigenous crops to encompass the two. Offer a sustainable source of nutrients that can supplement diet. That's my contribution on that. Then on your materials and method, there's a place while you are writing the species, you use capital L for the Lanatus, and you also use capital M for the Maxima. They should be small L and small m. I wouldn't know whether Prof has something to say. Prof Sigler.
Thank you, sir, for the observation. Thank you, sir.
Okay, you can. Okay.
They are special, yes.
Okay, no problem. Thank you very much. You can unshare. You unshare.
Okay,
now.
thank you, sir.
Thank you.
Thank you, sir.
At this point,
any one person?
Hello?
Hello.
Yes,
Good
sir.
afternoon.
Hello, sir. Hello, sir. Good afternoon, sir.
yes,
Dominic, sir.
do we have
Oyika Dominic.
Khadija? Khadija, are you ready?
Suleiman.
Who is speaking?
Oyika Dominic, sir.
Ready, sir.
Khadija Suleiman Jafar.
Okay, let's have you.
Okay, sir. Let me share my screen, sir.
Go ahead, go ahead. We can see you, but we have not seen your file. We can see you, we have not seen your file. We can see you, we have not seen your file. Hello? Khadija, where are you? You have muted yourself. Jafar, you have muted yourself. Unmute. Unmute yourself. Jafar, unmute. Unmute yourself. Okay, Jafar, we will keep trying. Who else is ready? Khadija.
Oyika Dominic, sir.
Huh?
Oyika Dominic, sir, is ready, sir.
Wow, Bello is ready, sir.
Hadiza, are you ready? Hadiza, are you ready? This one is Khadija. This one is Hadiza. Okay. Ohika, come on.
Sir? Okay, sir.
Come on.
Are you seeing the screen, sir?
Yes, we are seeing the screen. You have 5 minutes.
Okay, sir.
We are not seeing
My name
your
is-
title properly. What's your title?
Are you seeing it now, sir?
We are seeing it now. Okay.
Okay, sir. My name is Oyika Dominic Afamifuna from the University of Nigeria and Soka. I'm presenting on the topic Plasmid Studies and in vitro and in vivo study antioxidant property of methanol with DCM extract of stem of Euphorbia poison pass. My introduction.
Antioxidant damage is a term frequently used to imply random discriminate destruction to a wide range of macromolecules. Oxidative stress is generally defined as imbalance between the production of this oxidative stress with the body's ability to balance them. This reactive oxygen results from oxidation, which is a chemical reaction that involves the transfer of electron or oxygen or hydrogen from compounds to oxidizing agents, which result in free radicals that cause the damage to biological system. Antioxidants are groups of compounds that neutralize free radicals and reactive oxygen species in the cells. It is any substance when present in low concentration can substantially lower down or stop the oxidation of oxidizable substance. Traditionally, they have been employed to prevent oxidation in foods, but they are also quench free radicals and stop oxidation chains in vivo. Number of classification has been used to classify these antioxidant molecules, including natural, synthetic, primary, secondary, enzymatic, and non-enzymatic classifications. Present study is a chemical analysis which is mostly used in nutraceuticals or a nutrient to evaluate the nutritional values of food. But in pharmacognosy, they are also used to validate the quality and the purity of plant samples. Euphorbia poisonae is a broad group of flowering plants which grows on rocky and stony soil in the mountain wood and grassland. These plants are commonly found in the northern part of Nigeria. This is the picture of the plant I'm discussing about. Here is my methods and material. The experimental animal was obtained from Veterinary Medicine Department, University of Nigeria, Nsukka. Fresh stem of Euphorbia poisonae was obtained from Plateau State in Jos. Then methanol with DCM, some reagents and equipment. My plant collection, sampling preparation, that is cutting the stems, drying and washing, then grinding. After which plasmid study was carried out, that is the ash value, the acid insoluble value, the water soluble acid value, the moisture content before extraction was done to evaluate the extractive value and percentage yield. Before the in vivo antioxidant study was done, acute toxicity of the test was done before then. Then after which in the in vivo antioxidant study, I did SOD, CAT, and MDA. Then the in vivo aspect, I did nitric oxide radical salvageable properties. I did reducing power, then with DPPH. Then this is the table of my results for the extractive value of the plant. This table corresponds with the previous research works. Then this is the ash values for the total ash, the acid insoluble ash, and water soluble ash value. These values are presented in numbers. Then here's my result for moisture content, which is 8.8%. Here is the result for the DPPH. The IC50 is 468.3 when compared to the ascorbic acid, which was 97.02. This is the graph of the DPPH. This is the nitric oxide salvaging activity. Also comparing with the standard, which is ascorbic acid, it gives 228.01 against 6.06. This is also the graph. This is the result for total oxidative capacity, which is 201 against the 450 against the 43.03. This is also the graph. This results for reducing power. No, there was no IC50 calculated for this. We just compare the concentration and the readings from SV.
Please, conclusion.
Okay, in conclusion, at the end it was found that this plant exhibits antioxidant property when compared with the ascorbic acid, which is the standard. Also in vivo, it shows a more active antioxidant properties. And when compared with other previous research, the moisture content, which is from a part of the former study, and ash value was in line with what has been previously carried out. Thank you for listening.
Okay. Thank you. Please leave this your last slide on. Can you see it? No.
Last slide.
Yeah, your last slide. No, is
this one?
No, your last slide. Last one.
The first one.
Last one, the very last slide.
Okay, this.
Okay, no, the next one.
This.
Next one. No. Go the other way. Forward, not backward.
This. Okay.
Forward. Why now? Why are you doing like this?
So I'm using phone to present, that's why. I don't know the particular slide you're talking about, so please, sir.
The slide
pardon me.
Where you say thank you for listening.
Okay, sir.
Thank you for your time. Yes. I think
okay, sir. Sorry, sir.
You see it, ba?
Yes, sir.
Are you seeing it well?
Yes, I'm seeing it, sir.
Okay. No, it seems you are not seeing it well.
I'm seeing it, sir.
Okay.
Okay. I'm so sorry, sir.
Okay.
I was having an exam, so I prepared under exam pressure.
Yeah. When you are not in company of your friends, you know.
Okay, sir.
Yeah, this kind of thing may be costly for you. Do you understand?
Okay, sir. Yes, sir.
So pay attention and be careful next time. All right?
Thank you, sir.
Okay.
Thank you, sir.
Show us the picture of the plant.
Okay, sir.
Yeah.
This is the picture, sir.
Okay. Yeah. All right. Any question for the speaker? Okay. I have to thank you. There is no question for you. Thank you for your presentation.
Thank you so much, sir.
Yeah. Please unshare yourself. Thank you. Okay. Now, who is ready?
Yes, Alapha is ready, sir.
Suleiman Jafar, Khadija Suleiman Jafar. How far? Are you ready?
Yes, sir.
Okay. Go ahead. Go ahead. Share your file.
Okay. Okay, sir.
Okay, let me add. Are you sharing? Are you sharing your slide?
Yes, I've shared my slides, sir.
We are not seeing it yet.
Let me recheck again.
Let me suggest that maybe you check your network, or even check your data. Do you understand?
Okay, sir.
Just check. We will still come back to
Thank
you.
you. Okay, thank you, sir.
All right. Who else is ready?
I'm ready, sir.
Hadiza, or who?
Munira to Umar Shehu.
Who?
Munira to Umar Shehu.
I don't get the name.
Munira to Umar Shehu.
Munira to. Go ahead.
Yes. Okay, thank you, sir.
Share your file.
Okay. Good morning, good afternoon, everyone.
Yeah, put it on presentation mode. Put it on presentation mode. We are seeing it. We are seeing it, but it's too tiny.
Okay. It's because I'm using my phone, sir. Maybe that's why you can't see it well.
Okay. You want to present it like this?
Yes, because my system is not with me.
Okay.
If it's okay.
Go ahead. You have 5 minutes.
Okay. Good afternoon, everyone. My name is Munira Umar Shehu. I'm here to present my paper on evaluation of bioaccumulation and translocation of heavy metals by Moringa oleifera grown in soil obtained from the mechanic workshop Sokoto Metropolis. Introduction. The improper disposal of spent engine oil into soil significantly affects its structure and fertility, ultimately leading to poor plant growth. Phytoremediation is the use of plants to remediate contaminated soil. It is increasingly recognized as a sustainable, cost-effective, and environmentally friendly strategy, particularly in polluted agricultural and industrial areas. The aims and objectives. The study aimed to evaluate phytoremediation potential of Moringa oleifera in remediating the selected contaminated soil. The specific objectives are to evaluate the bioaccumulation factors of the plant species, evaluate the translocation factors of the plant species. Methodology. A completely randomized design was employed. Weighing balance calibrated in kilogram was used to weigh 5.
Hello, hello, are you there? Hello? Hello? Are you there? Hello? It's like we have lost her. Hello? Okay, please, studio, take control.
My brother.
Studio is everything is remote. His own is to focus the camera. Our own beam. They are beaming themselves. This one is beaming us. Sorry, we lost Munira. Who else is ready? Who is ready?
Okay, I am ready, sir.
Hawa Bello is ready.
Go ahead.
Alasan.
Bello.
Bello no tumo ba yo.
Bello.
Yes, sir.
Yeah, go ahead. Share your file. Yes, we are waiting for you. Yes, we are waiting for you.
Yes, sir. What will
Share your file.
Yes, sir.
Okay, it's coming.
Yes,
Yes.
sir. My name is. Good afternoon, everyone. My name is Awa Ubenda Abubakar.
Okay, but preferable in presentation mode. But if not, you can go ahead. Okay. Yeah,
Yes,
you are
sir.
good to go now. You have 5
Okay, sir.
minutes.
Okay, sir.
Yeah.
Medicinal values. My topic is medicinal values of some selected plants found in Mayama local government area of Kebbi State. Introduction. Nigeria is richly endowed with indigenous plants which are used in herbal medicine to cure diseases and heal injuries. The World Health Organization has estimated that up to about 80% of the world population rely on plants for their primary health care, while in Nigeria, the World Health Organization survey estimated that up to about 75% of the population patronize traditional medicine. Statement of the problem. The major challenges to the management of the human ailments are diverse and formidable. The conventional drugs available used for the treatment of human ailments in the healthcare system are extensive. As such, research for help in the medicinal plants which are accessible and cheap for the treatment of human ailments is worthy of scientific investigation. Therefore, a little research work on medicinal plants used to treat human ailments available in Mayama local government area of Kebbi State was made. Then the justification of the research. The people of Mayama local government area of Kebbi State, Nigeria, utilize medicinal plants for their primary healthcare needs. Some of these plants and their parts collected from various habitats are used either singly or in combination for the treatment of human ailments. Aims and objectives of the study. The aim of this research was to investigate and document the medicinal values of some selected plants in Mayama local government area of Kebbi State. The main objectives of this research are to document the medicinal plants and traditional knowledge of local communities on indigenous plants and their medicinal values. Study area. Or let me just methodology. Information about the values of some selected medicinal plant was obtained by conducting oral interview with the local medicinal practitioners and elderly members of the society whom have knowledge on the value of medicinal plants. Major information was gathered from these practitioners, although not all were given by them. They also gave brief details on preparation of medicine from plant, method of administration and dosage, collection and preservation of plants. Plant samples were collected from the field and were dried and compressed in newspapers. Newspapers were changed daily until they remained dried after compression through the interview. Local plant names, plant parts, method of preparation and administration were considered and recorded. Data collection. Data were collected between June and July of the year 2024. Each herbal practitioner was interviewed once. Information collected from these herbal practitioners include demographic of interviews, educational background, years of experience, information on medicinal plants like name of the plants, local names, and how they used to prepare the medicines. Result. Data presentation, demographic of the interviews. Or let me just skip this one. Like their ages, their gender, and total number of people that were interviewed. Educational levels were also considered. Just as you see, some plants like Anonaceae, Mimosacea, and others were used. like how they were used to cure the ailments, part of the plant that are used, method of preparations, how it is administered, all were shown. Then the recommendation and conclusion. The major challenges of the traditional medicine practitioner in study area are education, transportation, wild preservation, and dosage measurement are serious problems. State government should, as matter of urgency, look into these problems and find a lasting solution to them. There should be a policy that will provide minimal education for both the herbalists and farmers, as this will encourage them to practice their profession effectively. Thank you so much, sir. That's it.
Thank you so much for your interesting presentation. Let me ask, which one of these plants have you encountered personally?
Personal?
Yes.
The Acantharis aspera.
Yes.
There is Malaysia, Acanadecia. There is balanite.
Okay.
There is citrus, like orange.
Okay. You have encountered them. I mean, you have used them medicinally.
Yes.
Oh, okay. So you can vouch for them.
You said, sir?
You can vouch for them.
Yes, sir.
Okay.
The Prof, sir, I want to observe that the haslets are too chalked up. They don't look like PowerPoints.
Okay, sir.
Good observation.
Hello, sir, Prof, sir.
Yeah, we are hearing you. That's a good observation.
Hello, Prof, sir.
Yes.
In future presentations, the slides should be
in
PowerPoints, not
okay, sir.
No slide. Okay.
Which slide?
This is not PowerPoint. That's just the message. All right.
Okay, sir.
In your work, the plants listed, some of them you cited the authority, some you never cited the authority. I hope you understand what I'm saying. Hello?
Yes, sir. Hello, sir. I'm hearing you, sir.
Maybe because of space, some of at the level of the scientific name of your plant, some of them you cited the number second one, Acacia nilotica Lin. The first one,
yes, sir.
I wouldn't know where you got the PAR. Maybe you wanted to write VAR, maybe variety, so. And that's not Senegalensis. Yes. Then that second one,
okay.
I would have preferred that
yes,
you just write Mimosoideae fabaceae. I prefer that one instead of elevating the mimosa group to a whole family. Then your toothache, maybe treatment cured. The spelling is not correct. Then I just want to ask you a question. Under your materials and method, you said why the plants were collected, that you change the newspapers every day. Why? I hope
so that they will not.
You said newspaper were changed daily until they remain dried. Why?
So that they will not, the plant collected will not spoil.
Spoiled by. You can answer it now. I just wanted you to answer it. Spoilage by maybe fungal, waterlogged problem, so that they now get fungal infestation.
Okay, sir.
Good. Any other question? Do we have anybody online that want to make an observation? Anybody?
I'm ready. Akimbote.
Okay. We'll come to you. All
Sir,
right.
I am back again.
Thank you for your presentation. You can
Okay,
step
sir.
down now. Okay.
Okay, sir.
Yeah, who is ready?
Sir, my
Yes,
network is back now.
Akimbote is ready. Yeah.
Okay. Khadija,
Hello.
Sir.
are you ready?
Hello.
Yes.
Yes, I'm ready.
You want to try again?
Yes, it's my screen showing, sir.
Okay.
Please, sir, is the screen showing?
Are you sharing? Are you sharing?
Yes.
We have not seen it.
Yes, I'm sharing. Is it showing?
Not yet.
I don't know.
Okay. Still not yet sharing. So let's go to Akibot. Where are you?
Okay, sir.
Okay.
I'm ready.
Go ahead. Share your file.
All right, sir.
Okay. Are you sharing?
Yes, sir. I'm set now.
We have not seen it.
So can you see my screen, sir?
We have not seen it. We have not seen your screen.
Okay.
We have not seen your screen.
Okay, sir. So can you see it now, sir?
Not seen, not seen. Who else is
Okay,
ready?
sir.
Sir, my network is back now.
Who?
Munira to Umar Shehu, the one who presented earlier.
You want to come again?
Yes, sir.
We are struggling to get other people to present. Oh. Please. Let me get other people to present.
I'm live if I can present now, sir.
So who is there that is ready?
I'm here,
I
sir.
am, sir.
I'm also ready, Prof.
What's your name?
Garba Hani Dalladi.
Okay, Garba, let's hear you.
Okay. Just a minute.
So Garba Liu,
Checking.
we are not seeing.
Yes, checking the file. After sharing, I have to check the.
We have not seen you. It's not.
I think I'm on, sir.
Okay. Who do we have?
Dalladi Garba Hani.
Dalladi Garba Hani.
Yes, sir.
Go ahead. Go ahead. You have 5 minutes.
Okay. Characterization of rhizosphere nematodes of Solanum lycophyllum and Capsicum annum for the BOSON 31st BOSON 2025. these two vegetables happen to be some of the staple products worldwide, and then aleru is not an exception. And then the farmers here produce it both as a cash crop and for food consumption. They are how the nutritional and medicinal aspects of these two vegetables cannot be overemphasized because they have been proven to be important in the prevention of chronic diseases such as heart disease, cancer, diabetes, and the rest of them. They also have they serve as the supplements for macronutrient deficiencies in mostly used in the less developed countries of the world. Now, the problem of the cultivation of these vegetables, one of them is the nematode infestation. They have. So, in situations where these vegetables are cultivated, sometimes you find them in environments that are not healthy, or maybe farmer practices that carry um, kitchen waste and other household waste in order to serve as fertilizers as for the production of these products, and then they are prone to nematode infestation. So samples were collected randomly at various locations, 4 locations, and then the soils were collected from 0 to 15 cm depth, and then homogenized. That is, they were now produced as composite samples. They were labeled and taken to the botany laboratory for analysis, extraction. The trace extraction method was used for extraction of the nematodes, and then composed of double fly sandwich in a pair of plastic sieves placed in water bowl that has 500 ml of water. And it was allowed 24 hours. Then sieve was now gently lifted off, in which case the nematodes would have settled underground. Now, after 5 hours, the nematodes were settled, and then identification started. The compound microscope was used first at 100 magnification before now moving to 400, so as to look at the features of the nematodes. Where in the head we have the stylet, the lips and aphids, while the body, the cuticle, anus, and lateral lines. Then the tail shapes were also used to determine the types of nematodes that are there. So the nematodes,
Yes, yes. Where are you? Speak on, speak on. Speak on. Speak on. We are not hearing you. Okay. Studio, please take control. Studio, take control. Thank you. Sorry, we cannot continue with you. We are not hearing you again.
Hello, sir. Can you hear me?
Yes.
Hello.
Yes, we are hearing you.
I'm ready to present, sir.
Okay. What's your name?
My name is Bawa Saidu from Osumandanford University, Sokoto.
Yes.
I'm sharing my screen right away. Can you see my screen, sir?
Not yet. Okay, okay. Bawa Saidu. Go ahead. You have
Yes,
5
sir.
minutes.
Okay, thank you, sir. Um, my name is Bawa Saidu, as earlier stated. A PG student from the Department of Plant Science, Usman Danfodio University, Sokoto. I'm here to present a paper titled Phytochemical Analysis, Anticoagulant and Anti-Inflammatory Activity of Acacia seberana stembank. Introduction. Now, natural products derived from various medicinal plant parts have long been used in medicinal, pharmaceutical, and agricultural applications. Natural products exhibit superior efficacy and safety profile in comparison to its synthetic counterpart. Medicinal plants offers therapeutic properties through the secondary metabolites they possess, which also aids medicinal development. Why this research? The reason for this research, one is about 1/3 of modern medicine comes from plant-based sources. This highlights the importance of medicinal plants in pharmaceutical development. Some studies by Ngafo reported the presence of some phytochemicals and biological activities in these plants, Acacia seberana, such as the antioxidants and so on. This plant is traditionally used to treat wound, according to a survey done by Matthias in the year 2023. But little study has investigated the biological composition, the phytochemical composition and the biological activities in regards to this bioassay. Justification. Now, 80% of the world's population depends on plant-based medicine as their primary source of health care, according to the World Health Organization. Medicinal plants depletion is one of the drawbacks that is affecting medicinal plants worldwide, which is caused by overexploitation. But biotechnology offers promising solutions through tissue culture and also genetic engineering. One of the justification is the high cost and potential side effects of this orthodox medicine, which have made people to look elsewhere for a safer, more accessible, and more affordable source of medicine. Now, these are the aims and objectives of these studies. The aim is to evaluate the phytochemical composition, anticoagulant, and anti-inflammatory activity of the Acacia seberana stembank. Below are some of the specific objectives listed, which includes the evaluating the phytochemical constituent, the anticoagulant constituent, and the anti-inflammatory potential of the crude extract and fractions of Acacia seberana stembank, and then to evaluate the toxicity of the crude extract of the Acacia seberana stembank. Here listed are some of the materials we made use of, such as oven, mortar, pestle, separating funnel, centrifuge, stopwatch, syringe and needle, etc. Solvents that we made use of are the methanol, N-hexane, ethyl acetate, N-butanol, and distilled water for the fractionation procedure. The reagent we made use of are the trisodium citrate, the PT-assay reagent, APTT-assay reagent, and calcium chloride. Sample collection, identification, and authentication. The sample was collected at Usman Danfodio University, Sokoto, the environs, and the identification and authentication was done at the Department of Plant Science, Habiram, and a voucher number was issued. This is how the sample was prepared. The stembank was oven dried after collection and pulverized using pestle and mortar, and the powdered sample was stored in an airtight container before its use. Now, the plant extraction and bioassay guided fractionation procedure was carried out using a method described by Ayodele, while the phytochemical evaluation was carried out using Fourier transformed infrared spectroscopy analysis, FTIR, and also a procedure described by Vishnu for the authentication of the functional group found in the FTI spectrum. The acute toxicity of the crude extract was determined in Wistat using method described by Locke and also adopted by Adeinka, while the clotting profile, APTT and PT assay was determined using standardized protocol as described by Zhang. The anti-inflammatory potential of the crude extract and fraction of Acacia seberana stembank was evaluated using egg albumin inflammatory model adopted by Enechi. This here is the result after the extraction procedure and also the concentration of the extract. We obtain a sticky dark brown solid residue of 212 gram, which is 15.9% weight per water of the previously used sample, with a mild sweet smell and was obtained from the 1,333 gram of the sample used. Now, the phytochemical evaluation revealed the presence of some functional group, which were later identified through the other process as terpenoid, saponin, flavonoid, tannin, quinone, and glycoside. The acute toxicity study of the crude extract revealed that the oral median liter dose of the extract was greater than 5,000 milligram per kg. Now, here is a bar chart showing the performance of the crude extract and some of the fraction. As we can see, the bars are of different colors, which signifies the crude extract, and the three fractions that we have, all of them are of different concentration in comparison to a well-known anticoagulant drug, heparin. Here also is the APTT performance of the bar chart and of the crude extract and the fraction. The bars are of different color, which signifies the crude extract and the different fractions that we have. Also in comparison to the well-known anticoagulant drug, heparin. Here is the percentage inhibition of the crude extract and fraction of Acacia severana stem bark, also showing different colors under different concentration in comparison to a well-known anti-inflammatory drug, diclofenac. In conclusion, the crude extract of Acacia severana was found to be relatively safe and non-toxic at 5,000 milligram per kg, and the crude extract and fraction also showed dose-dependent anticoagulant and anti-inflammatory activities, with the ethyl acetate fraction of Acacia severana exhibiting the strongest effect on PT and APTT assay, while for the anti-inflammatory model, the crude extract, the crude aqueous methanolic extract of Acacia severana and the ethyl acetate fraction of Acacia severana had the highest percentage at 25.58% and 28.72% at the highest dose of 100 milligram per kg in comparison to 25.79% of 50% diclofenac. Recommendation. Now, further research is needed to isolate and identify those bioactive compounds present in the stem bark to evaluate them to see which of these compound is responsible for this bioassay. Secondly, the mechanism of action of the crude extract and fraction of Acacia severana also need to be evaluated. And then lastly, the therapeutic potential of this crude extract and fraction should be evaluated in clinical trial to ascertain its safety and toxicity profile. Thank you for listening.
Thank you for your presentation. Any question?
Please, can we see your title? Your title?
Okay.
Your DC is Dicandoli. So it's not a matter of D full stop C. It should just be DC.
Okay.
So it's not a matter of D full stop C. It's just DC.
Okay, sir.
Okay. Any further question? Is there anybody online that want to ask?
Yes, sir.
Okay.
Akinbote is ready, sir.
Oh, I'm asking for questions. Yeah. Okay, Bawa Seidu, you can step down now.
Thank you very much, sir.
Okay. It has been a long day, all of you online. Let me take this moment to appreciate your your, how will I put it, dedication and your deep interest in these presentations. Since the beginning, we have had scores and scores of people online joining to present and to contribute. So we really deeply appreciate you for your deep interest. I have to unfortunately tell you that we have space for just one or two presentations at most. Thus, if the first one can save time. Before we take a break and continue with other programs. So let me request to know, I know that Khadija Suleiman Jafar has been struggling to link up and present. I will give you a chance.
I am also available to complete my presentation, Prof. The network cut off while I was at the result level.
If we give you chance, you know, go and start from the beginning again. No?
Yes, I cannot.
You will just link up where you stop. All right. So who goes first? Khadija, you want to try? You want to try again? Khadija Suleiman Jafar. Unmute yourself. Unmute yourself. Do you want to try again?
Yes, I want to try, sir.
Okay, so I wish you good luck. Go ahead.
Thank you, sir.
Yeah.
Sir, is the screen showing?
It's coming. It's coming.
Okay.
But we have not seen it yet.
Okay.
Okay, it's coming. It's coming. We have seen some part of it.
Yeah, okay.
It's still coming. It's still coming.
Okay. Okay.
Let me just ask you to go ahead. Since we are hearing your voice, beautiful. It's on now, but can you try? Okay, beautiful. Go ahead. Go ahead. You have 5 minutes. Yeah.
Good afternoon, ladies and gentlemen. I am by name Khadija Suleiman Jafar, presenting a paper on oilseed extraction and characterization of calabash grown in Gumi local government area of Zamfara State from Department of Biological Sciences, Federal University, Gusau. By means of introduction, Lagenaria caesarearia, commonly known as calabash, white bud gourd, bottle gourd, or long melon are vine that are mainly cultivated because of their fruits. Calabash is classified to the kingdom plantae. It is one of the first cultivated plant in the world, grown not for food but as a container. It can be used as vegetable when harvested at a young early stage, or used as utensils, pipe and bottle when harvested at a mature stage and dry. Oil is used in generic sense to describe all substances that are greasy or oily fluid at room temperature. Olawefor et al. 2012 and Sani et al. 2013 describe calabash as a climbing ornamental plant grown as a shrub with its fruiting hanging on a flat bed, which is usually harvested between 90 to 120 days after planting. And majority use it in rural settlement as a container and storage vessel. The calabash fruit has light green color with smooth skin and white flesh. Calabash grown in different sizes and shapes such as small and bottle-shaped, huge and round, slim and widened, longer than a meter. Calabash gourd are varieties that are round in shape, and it is mostly cultivated in the tropical region of Africa as Nigeria. Statement of the research problem. Vegetable oil play a significant role in food, cosmetic, and pharmaceutical industries, with increasing global demand prompting the exploration of utilized oilseed crop. L. caesareae, which is a member of the family Cucurbitaceae, is traditionally cultivated for its fruit shell as a container, but its seed remain unexploited despite being rich in oil and nutrients. Previous studies indicate that calabash seed contain appreciable amount of lipid, protein, and essential fatty acid, which highlights their potential as an alternative oil source. However, limited scientific data exists on the efficient extraction method, phytochemical properties, and nutritional quality of oil derived from the calabash seed. The lack of comprehensive characterization has also hindered the industrial application of calabash seed oil, particularly when compared to more common oils such as that of groundnut, soya beans, and palm oil. Therefore, there is need to extract, analyze, and characterize oil from the seed of calabash. Objectives of the study: to extract the oil present in the seed of calabash plant, to characterize the oil extracted from the calabash plant. Materials and method: the study was carried out in the Department of Biochemistry of Faculty of Science, Federal University, Gusau. The mean annual temperature is about 7.315 degrees Fahrenheit. Sample collection and preparation: the fruit of calabash was collected at random from different farms in Gumi local government area of Zamfara State. The fruits were identified by a botanist from the herbarium unit of Department of Biological Sciences, Federal University, Gusau, after which the seed was removed and washed. The seed were air dried, broken mechanically, and separated from the spongy part for 7 days, and then melt into powder using mortar and pestle, after which it was sieved for extraction of oil. 60 grams of 2 millimeter of calabash seed was placed in a chamber of solid apparatus and extracted with petroleum ether at 80 degrees Celsius for 2 hours. The same procedure was repeated with a sample of 4 millimeter and 6 millimeter particle size respectively. Characterization of oil extraction: here the pH determination was carried out. Determination of saponification value was also carried out. For specific gravity, 20 grams of extracted and refined head oil was mainly in adipose tissue and had to be meshed. This volume was determined and the volume of water was measured using the following formula. For refractive index, Abel refractometer was used to determine the refractive index of the extracted oil. For boiling point determination, 80 milliliters of the extract.
Go to your results.
Okay. Table 1 shows the results for the phytochemical properties of the extracted calabash oil, where we have a pH of 6.6, acid value of 2.25, and the rest. Results and discussion: the pH value indicated that oil is slightly acidic, indicating the presence of small amount of free fatty acid. The acid value of 2.256 milligram per gram obtained in the study is much higher than 0.47 milligram per gram reported by Wara et al. 2016. However, it is comparable to 2.02 milligram per gram reported by Denjuma and Dandagu 2009. The acid value of the oil is used as a general indicator of the condition and edibility of the oil. According to Arimu 2006, acid value gives the indication of the quality of fatty acid in oil for edibility of an oil. and suitability for use in the paint and soap industry. The specific gravity, which is a measure of the amount of alkalinity required to break down the triglyceride in oil into soap and glycerol, and the specific gravity of calabash oil was 0.9, which conformed the standard value of less than 1.0. The average yield of the oil was found to be 90%, which could be attributed to the high moisture content in the seed, as the seed was not optimally dried and maximum extraction period could not be used due to intermittent power supply. Ibrahim et al. 2016 also reported that calabash seed oil yield of 39.3 by mechanical press, while Sokoto et al. 2013 and Uwana et al. 2018 from their study reported percentage oil yield of
You have
36.7%.
1 minute.
Conclusion: based on the result of analysis conducted, it can be concluded that extraction of oil from calabash seed could reduce the pressure and overdependence of available seed used for vegetable oil extraction for human consumption. It will also boost soap production and supplement the oil base of biodiesel production and lubricating oil, as it has a good quality profile with low acidity and high saponification value. Recommendation: as calabash oil is rich in fatty acid, antioxidant, anti-inflammatory and other nutritive components, it is recommended that more research on calabash oil properties and potential application should be conducted to uncover new uses and benefit. These are some of the references cited. Thank you for listening.
Thank you too for your enduring patience. Any questions
Thank you,
from
sir.
the audience? Yeah.
Thank you very much for the presentation. I hope you can still hear. I hope you are hearing me. Maybe she has left.
Yes, I can hear you, sir.
You need to edit your work more. Like this, thank you for listening. The end
Okay,
should
sir.
go. Then you wrote conclusions. It should just be conclusion. Then your title.
No, sir, because I have two
Let me just say
conclusions.
all what I have to say first. Your title.
Okay.
I hope you are
Okay.
aware of the calabash tree. So I think I would have preferred that you just say bottle guard from the title, because there is
Okay,
a
sir.
calabash tree,
Okay,
and
sir.
this is a climber calabash. So maybe characterization of bottle guard, GOURD. Then your introductory aspect. Your first introduction something, you really, there is a place ahead. That second bullet, you went off. I wouldn't know your mood when you are writing that second bullet. I hope you can see it yourself. Say calabash is classified to the kingdom of plantain. It should be capital.
Yeah, kingdom plantain.
It should be capital P.
Yes.
Division of.
Yes, I can.
Yes, division of maglonophyta. That one is not correct.
M capital. Okay.
Class of is MA. GNO hair. If you look at that class, it's MAGNIO. Order of Cucurbitaceae, family Cucurbitaceae. Then genus, the Lagenaria should be capital L. Then the species should be italicized. Lagenaria, it should be italicized. So you edit the work. It's a solid work, but please edit it better than this. Thank you very much.
Okay, thank you, sir. Thank you very much for your contribution.
Any other contribution? I'm just curious to know how feasible it is to gather seed from these plants in enough quantities to be able to produce reasonable amounts of oil. Is it a feasible venture?
So this is because of Gumi local government area of Zamfara state are known for cultivating this calabash. So
there is potential
that's why
there.
yes.
In addition, like she said, that environment is a highly it's a high calabash producing area. If you are passing by, you will see the calabash in thousands. And then the calabash itself, inside it, there are so many seeds. It has so many seeds. It is a very large fruit that it produces. So it's very, very possible to get these seeds in large amounts. So.
Okay. So thank you.
Yes.
All right. Because of time, we'll ask. Thank you for your presentation, and please.
Thank you very much,
Thank
sir.
you. All right.
Thank you, sir.
Last but not the least, who is that again?
Pastor is ready, sir.
Who?
Akimote.
No, we have only space for one person, and I've already promised. Who is to conclude his talk?
That is me, sir.
Okay, can you conclude it in 1 minute?
Yeah. So in conclusion, the research found, though it found some nematodes, but luckily enough, there is no any pathogenic nematode, especially to humans, that was isolated from the environment. So it means that the vegetables that are being produced in that environment are safe for consumption.
Okay.
And that, and I think it's very important in recommendation that the farmers should be more enlightened to make sure that they guard against any during production. The manures they're taking to the farms should be free from contaminations or from areas of maybe sewage disposal and the rest of them. Thank you very much, sir.
Okay. So the farmers are relatively safe from
Yes, it shows that yes, the practices of carrying the manure is safe to the farms. Yes.
Okay. All
So
right.
it's free of nematodes. I can't say about other microorganisms, but as per nematodes, it is safe.
Okay. It's good to know that. Very reassuring because just look at it, pepper. Who can do without pepper?
Yes.
So
nobody said it.
Thank you for your patience and for your. Okay.
Just a minor. Your Hanum is ANNWM. You kept on repeating it that way. ANNWM.
Capsicum annum. Okay, the capsicum annum.
Okay. So thank you very much.
Okay. Okay.
So for saving time, I think we can ask Aki to please share yourself.
Yes.
So do we still have Aki online?
Yes, sir.
Okay. 2. You have only 4 minutes. But we'll listen to you. Go ahead.
Thank you, sir.
Okay.
So, sir, can you see my screen now?
We are not seeing your screen, but I think you should just speak on. If you have the material, speak on.
Okay, thank you very much, sir.
Okay.
So my name. Okay. So my name is Alasan Daniel, and I'm just using Akimbo's device.
Go ahead.
So my presentation titled "Comparative Studies of Proximate Content of Fresh and Stock Kukuru Cassava Fermented Flour." Introduction. Nigeria population is about 217 million,
See,
according to Nigeria.
I advise that you go to your results data.
Okay, sir.
Because of time.
Okay, sir.
Okay.
Okay, sir. So, pupuru is one of those products we can get from cassava. So, result. What I try to do is to just do the proximate analysis of fresh and stored pupuru. So I look at the protein content, the percentage of fiber, moisture, and ash, fiber, and carbohydrate. So result, about 5 pupuru producing location sample were collected from 5 producing towns, and they were analyzed for those proximate content. For Odiaye, the fresh sample, the result, Odiaye, there is no significant difference between the fresh and the stored one, and we have Ore. So, looking at the moisture content of all the pupuru sample collected from Odiaye, Ore, Irele, Okitipupa, and Igodigo in Ilaje or Ilaje area in Ondo State, there is a significant difference from all of them except Irele and Igodigo. Why the crude protein only varies from Odiaye, Ore, Igodigo, the Irele, and Okitipupa, they are the same. Crude fat varies significantly from each location, and the same thing with crude fiber and ash. It is also noted that the carbohydrate content of this product is really high. The third sample, there is a significant difference from the moisture content, and the crude protein too has had a significant difference from all the locations except for Irele and Okitipupa. So for crude fiber, there is no significant difference between Irele and Okitipupa and Igodigo, but Odiaye and Ore had significant difference. Also, in their carbohydrate content, it is really high. So this is not far from the fact that this product is from cassava, so the carbohydrate is high. Discussion. I noticed that the moisture content increases with storage time. This is in agreement with Igbeka, who reported earlier that microbial load of pupuru flour over the storage period of time increased. And Idowu reported the same thing, that high moisture uptake and greater vitamin C and color loss in okra powdered that were stored. More so, the moisture content of pupuru increased with different packaging material. This was reported by Daramola Ito in 2010 and Booth in 2004. The increase in carbohydrate content, even though it's generally high, but decreases with storage time. This is also reported by Jonathan Ito in 2013, which may
Hello. Are you hearing us? Hello?
I think his network is not.
Go ahead.
Sorry, okay. This, of course, might be as a result of microbial degradation of nutritive substance found in the pupuru that the
Hello.
Yes, sir. Can you hear me, sir?
Yeah.
It's just the network.
Your line is ceasing.
Oh, sorry, sir. It should be fine now.
But
So the decrease in the crude fiber and the crude protein, the crude fat in ash must have been due to the fact that the fungi use up the content of the growth parameter and the substrate. This is all corroborated with the finding of Jonathan when he also carried out some research
So
on
you have
Galicia
1
and
minute.
Onula.
Just 1 minute
Okay, sir.
left. We'll go to
Okay, sir.
conclusion. Yeah.
So, conclusion. Carbohydrate was more for both fresh and stock pupuru, but poor source of other proximate constituent. You know, this pupuru food material is gotten from cassava, so it is understandable that the carbohydrate is high, but other proximate material like protein, crude fat, crude fiber, and ash, they were really, really small. So moisture content increases with storage, and fresh products have better proximate constituent. Recommendation: it is better to eat fresh pupuru, and if it must be stored, the length of duration should not be too long. Diet in pupuru should be accompanied with rich source of vitamins, with this oil of vegetables and protein. The process of producing pupuru, the location, the material for storage also plays significant role in moisture that the pupuru will take if it's going to be stored. Then storage of foodstuff like pupuru for a very long time should be discouraged.
We said 1 minute, 1 minute, 1 minute.
Okay.
So thank
All
you,
right, sir.
thank you, thank you for your deep interest and determination. You seem not to be aware of the amount of confusion you created with this your pupuru that sounds like guguru without taking care, you know, to take care of those that are not familiar. So, in fact, hearing that guguru is made from cassava is a cataclysmic, has created so much turmoil in some people, you know.
Okay.
So
I'm sorry for the pronunciation.
Except just to draw your attention to the fact that the potential exists to create confusion when you discuss this subject. So, ab initio, you should always try to make the distinction clear so that people can follow you. You have a question?
Thank you so much, the presenter. Well done. White. You are from UI. Actually, the Pupuru Prof said we really enjoy Pupuru in Ondo Town because I studied in Ondo Town and I was privileged to eat Pupuru. So it's a good food. Thank you for your contribution on our Pupuru with Foilegede in Ondo Town. Thank you. I enjoy it.
Yes, sir. Thank you, sir. Thank you, sir.
Hello. Any other?
Yes, sir.
Is there anybody online there that wants to contribute to this? Okay. Hello. Okay.
Yes, sir.
We want to give some time out for people to rest before we start the AGM. So, but is there anybody out there that need to make a presentation?
Okay, yes, I've not presented.
And you have been so patient online.
Yes, I've been waiting for
You are
hours.
definitely undeniable. So
Thank you.
go ahead, go ahead.
Okay, I'll share my screen. I'll be quick. Thank you.
Okay.
So thank you. Please, can you see my screen?
We can see you clearly.
You can see it now, yeah. It's okay.
Go ahead.
Okay.
You have
Okay.
4 minutes.
Okay, thank you. My name is Rejoice Anodo, and I'm a student from University of Nigeria, Nsukka, undergraduate student. So I'll be presenting. The topic of presenting is the phytochemical evaluation and anti-inflammatory activity of the methanol root extract of Momodica angusticephala hams from the family Cucurbitaceae. So inflammation is a defense mechanism in the human body and in other organisms that can become harmful at side effect. And to handle inflammation, there has been the development of synthetic drugs, mostly non-steroidal anti-inflammatory drugs like paracetamol and diclofenac. And these drugs most times come with
Rejoice, your volume
Yes.
is a bit low, so we are not hearing you clearly.
Okay, I'll speak louder. Can you hear me now?
Yeah, better now, better. Please
Okay,
speak louder.
Okay. So the development of synthetic drugs have been done over time to combat inflammation, and the use of these drugs have also caused side effects like cardiovascular diseases and gastrointestinal problems. But due to this, there's also been the need to create safer, cost-effective alternatives. And Momodica, traditionally in African ethnomedicine, it has been used for anti-inflammation, pain relief, and to handle cases like arthritis and osteoporosis. This research study was carried out to evaluate its scientific potential and to confirm if it can be used as a safer, cost-effective alternatives to synthetic drugs that are used to cure inflammation. So this is a visual representation of what the root tuber of Momodica looks like. This root tuber was gotten and it was dried. That's why it looks like this. So the objectives were to identify the phytochemical constituents in the methanol root extract of Momodica angusticephala and to evaluate its anti-inflammatory activity in marine species, specifically in albino wistarats, and to support the use of African ethnomedicine with fact-based research. This research was carried out with these steps. First, the root tubers were dried, were cut into little pieces. They were dried and they were grinded at a local milling station, and the extract was gotten with methanol. And after that, we carried out quantitative and qualitative phytochemical screening. And after that, there was animal study conducted, and it was done in 2 sections: the acute model and the subchronic model. And while this was carried out, we tested for the odema which was induced in the murine species. So the results of the research are as thus. The phytochemicals found in the methanol root extract include phenolics, saponins, alkaloids, tannins, flavonoids, and terpenoids. From the results shown, the phenolics are the highest phytochemicals found in the methanol root extract, and carbohydrates were the had the highest concentration of carbohydrates were the highest macromolecules found in the root extract. So this is a table showing the amount of phytochemicals found in the root extract. As you can see, phenolics is the highest and carbohydrates is the highest macromolecule. So for the anti-inflammatory activity, the adenomyster rats were split in two groups. There were 40 rats, and in these two groups were split further into 5 groups each. So in both models, the rats were given varying doses of the plant extract. The first group were given the plant extract before it was induced with carrageenan to stimulate the oedema in the hind paw. So they were split according to their weight. So the first was 100, and the plant extract was given like 100, 200, and 400 mg. That's the plant extract. And then the fourth group, it was given 10 milligram of diclofenac, and then the negative control was given water. Then the second group, it was the subchronic. So in the second group, we used formalin, not carrageenan, and it was induced the same way in the right hind paw of the test subject, and we used the same method, the 100, 200, 400, and 10 and 5. The first 3 groups were the plant extract, the fourth group was the control drug, diclofenac, and then the fifth was negative control. So the results show that the plant extract was indeed very helpful in combating inflammation. And in the first group, the highest level they got to was 50% at 100 milligram per kg. And in the second group, the subchronic model, it was 57.89 percentage inhibition of the oedema at 200 mg per kg. So this is a visual representation of the table. As you can see, it happened, the acute model, it happened at a shorter interval. At the 3rd hour, we got the highest efficacy. And in the subchronic model, we got the highest efficacy on the second day at 200 mg. So from the research done, we concluded that the plant can be used as an anti-inflammatory agent, and the presence of the active constituents in the methanol root extract supports its anti-inflammatory activity, and the extract outperformed diclofenac in some measures, and it was more effective in the subchronic model than the acute model. So the conclusion of the research is thus: Omodica angustifolia is effective against inflammation and can serve as a natural alternative to synthetic non-steroidal anti-inflammatory drugs like diclofenac, and its potential can be explored further through bioactive compound isolation, safety profiling, and formulation. So I was assisted in the laboratory by Mr. Felix Wafo and Dr. Austin Okori, and I was supervised by Dr. Blessing Chigo. So thank you.
Okay, thank you so much for your beautiful presentation. Anyone in the audience that wants to ask a question?
Please, can we see your introduction?
Okay.
It's not okay. Did you ever describe the plants there? that maybe is a climber, the nature of the leaf, the nature of the seed, the nature of the fruit, very essential. Because from our own experience, I'm aware, and I believe you should be aware of Momodica charanta.
Yes, I am.
The most popular
I
one.
in my study.
I'm
Yes.
also aware of Momodica schizoides, Momodica foetida. But I'm hearing this Angutisepala for the very first time. I'm not even too sure that it's present in the flora of West Tropical Africa, of which I have the compilations. So why I'm raising that issue is that I hope you are aware of what we call citations of authority, because you are introducing the plant to us. Just like somebody say, Carica papaya, Carolus linus, lean. I hope you are understanding, you are flowing with what I'm saying.
Yes, I am.
You should have gone to the extent of providing us with the citation of authorities as those who cited or who named the plant. So that's my observation for now.
Okay, thank you. So can I point it out now?
Yeah, talk.
Okay, so the plant was, the authority is hams, it can be seen in the title, and the family is Cucurbitaceae, and it shares the same, it's in the same genus as Momodica carantia, as you pointed out, and it is a climber, and the common name is narrow sepal bitter lemon, and the name is from the morphology. It has narrow sepals, and the fruit looks like, I'm sorry there's not an image here to show you what it looks like, but the fruit looks like something like cucumber. That's where it gets a common name from.
Okay.
So it's in the same family as cucumber and the sponge plant.
Okay. Is it a native plant species, and
Yes,
where do you find it?
Well, the plant I used was gotten from a community in Enugu State. So it was found in Obukwa, in a forest in Obukwa. It's close to University of Nigeria and Nsukka. It's within the environment, so it's native here.
Okay. Actually, we need to, you can improve this work a lot by showing the image of the plant. The plant appears to be new to many of us, and so we are really intrigued. In fact, as you are speaking over there, some of us have had to Google to be sure that we are in good state. Do you understand that? So
yes,
many
sir,
of us are not aware of this plant, but kudos to you. You have brought it to our notice. And okay,
sorry. Yeah. You said the purpose is for anti-inflammation.
Anti-inflammatory activity, yes.
Then you are working on rats. Yes, rats.
Yes, I use Abinoptera.
So how did you induce the inflammation on the rats before testing the Momodica antigusepala?
Okay, like I explained, we used two different, we used carrageenan and formalin. We injected 0.1 ml of carrageenan in the right hind paw of the rat. So after some time, the rats developed visible swelling at their paws. So administering the plant extract and the control drug, diclofenac, the swelling reduced after some time.
Okay.
In both models.
Okay, I think we have a lot of students listening to you, and I think you displayed something that many can learn from. How did you simulate your bullet pointer? Your bullet
I did
pointer
not.
on the tables, your colored bullet pointer.
Okay, I did not use PowerPoint like the app. I used Canva. It's a different app for design, but it can present PowerPoint as well. I find it easier to use. So That's why I used it. It's quite easy to follow. You can like learn tutorials on YouTube. That's how I used it.
Okay, it came with the application.
Yes.
The facility came with the application.
Yes, sir.
Okay. So Rejoice, Abby, am I correct?
Yes, sir. Yes, my name is Rejoice.
Yeah. I think, thank you for your patience. And in the end, I think we enjoyed you greatly. You have contributed a lot. Thank you
Thank you,
so
sir.
much. So to everybody online, please, you can join for the AGM, but for now, we are taking a short break before the AGM starts. Thank you all for your contributions and your patient endurance.
Hello, my name is Enakiri Desiree, and I am a proud student at Benson Idahosa University. When it came to choosing a university, I wasn't just looking for a degree. I wanted a place that would sharpen my potentials and align with my values. Benson Idahosa University stood out as that place. Here, excellence isn't just expected, it is nurtured. Our lecturers are mentors who challenge us to think deeply, lead boldly, and grow consistently. What sets Benson Idahosa University apart is its foundation in faith. It's an environment that builds both competence and character. I'm not just gaining knowledge today, but I am being prepared to make impact, to lead, and to innovate. Benson Idahosa University is truly preparing me for global success.
I'm Dr. Femi Idahosa, President of Benson Idahosa University. Now at Benson Idahosa University, that's BIU, our message is clear, empowering you for global success, academically, socially, and spiritually. We are committed to raising a new generation of leaders. Once again, that's you. Young people equipped not just to succeed, but to transform the entire world. Our graduates are living proof, making impact across industries and continents. They inspire us every day. Now to our current and incoming students, you are the agents of change the world needs. We need your voice, your vision, and your values. And the time to take ownership of the future is now. Behind every student's success is our dedicated staff of mentors and educators who believe in each student's potential and work tirelessly to bring it out. Their impact is real. We are more than just a university. We are a launch pad for greatness. And with each passing year, I grow more confident that we are doing something extraordinary. Because at BIU, everything we do is focused on one thing: empowering you for global success.
Hello, my name is Joshua Tamunateli, and I'm a proud student here at Benson Idahosa University. I had my reservations about enrolling into any private institution due to how one can feel restricted. And to be honest, I was wrong. The decision to come here has turned out to be an incredible journey. Benson Idahosa University has helped me grow beyond just academics. Here, I found my voice, built strong friendships, and learned under lecturers who consistently challenge us to think critically and become innovative. I came into Benson Idahosa University knowing little or nothing about life, but now I am a student leader. BIU is definitely shaping me for something bigger than just having a degree in my field of study. It is setting me up for global success.
Hi, my name is Agari Ogenemaru Daniel. I'm a 200 level law student. I got to know about Benson Idahosa University from a friend of mine, and he told me about it, and I said, fine, let's try it. And since then, my experience has been very interesting. I got to join the choir, and I also got to be part of the student leadership body, and it has actually helped me to discover my potentials and more things I can do as a singer. If I was to make a choice to another university, I would definitely and gladly choose Benson Idahosa University.
One of the things that we have desired about BIU is to make BIU unique, innovative, and do something that is not done by other universities. The type of graduate we need in Nigeria is not somebody that will just graduate from the classroom, have a BS, and it ends there. No. We have believed that Benson Idahosa University will be another university where other ingredients of success in life can be put into students.
What differentiates a good editor from an editor is how creative you can piece those footage together and make sense out of it. The colorist is in charge of evoking emotions using color on screen. Some elements of film editing include cuts. You know what cuts are, like the footage itself. You have to cut it somewhere. You have to join it to another footage that you likely have cut somewhere. Pacing. Pacing is the overall feel of the film. Continuity is important. It should follow the way your normal life would be.
And I'm so happy to be here. This is the day 3, and I've learned so much from the first day to now. And presently, we have learned so much about storytelling, cinematography, and a lot of other variables in the entertainment industry.
From a level of knowing nothing, I know almost enough to film a good movie.
I've learned a lot of things from storytelling, script writing, and video editing. It has been so wonderful.
And God sends abundant life to you in the name of Jesus. From the word we heard today, we will give you abundant healing in Jesus' name. From the word we heard today, we will give you an abundance of His grace in the name of Jesus. I prophesy to you in the name of Jesus. Everything God has spoken, you will see come to pass in the name of Jesus. Grace goes before you this week. His power will speak for you this week in the name of Jesus. Abundant life shall pursue you. Abundant life shall find you. Abundant life shall be your portion in the name of God the Father, God the Son, and God the Holy Ghost. We pray. Amen.
Who am I that the highest king would welcome me? I was lost, but he brought me in. Oh, his love for me. Oh, his love for me. Who the sun sets free, oh, is free indeed. I'm a child of God, yes I am. Free at last, he has ransomed me, his grace runs deep. While
Breaking news. The home is under attack by stains, disease-causing germs, and so much more. An all-out war. But not for long, cause hypo.
God of creation, there at the start, before the beginning of time. With no point of reference, you spoke to the dark and fleshed out the wonder of life. And as you speak, 100 billion galaxies are born. In the vapor of your breath, the planets form. If the stars were made to worship so loud, I can see your heart in everything you've made. Every burning star, a signal fire of grace. If creation sings your praises so loud, so God of your promise, you don't speak in vain, no syllable empty, oh boy. For once you have spoken, nature and science follow the sound of your voice. And as you speak, 100 billion creatures catch your breath, evolving in pursuit of what you say. If it all chased down my heart through all of my failure and pride. On the hill you created, the light of the world, abandoned in darkness to die. And as you speak, 100 billion failures disappear. Well, you lost your life so I could find it here. If you left the grave behind you so alive, I can see your heart in everything you've done. Every part designed in a word of my God. If you gladly chose to surrender so alive, I can see your heart in billion different ways. Every precious one, a child you died to save. If you gave your life to love them so alive, like you would again 100 billion times, but one measure could amount to your desire. You're the one who never leaves the one behind. All these pieces, broken and scattered, in mercy gathered, mended and whole, empty-handed, but not forsaken. I've been set free. I've been set free. Amazing grace, how sweet the sound that saved a wretch like me. Ooh, I once was lost, but now I'm found. Was blind, but now I see. Oh, I can see it now. Oh, I can see the love in your eyes. Laying yourself down, raising up the broken to life. You take our failure, you take our weakness, you set your treasure in jars of clay. So take this heart, Lord, I'll be your vessel, the world to see your life in me. Oh, amazing grace, how sweet the sound that saved a wretch like me. Ooh, oh, yourself down, raising up the broken to life. Hope be my anthem, Lord, when the world has fallen apart, you stand beside me. Give me a song in the night, and Jesus, I need you every moment. I need you here now. This grace, Lord, heart sing out your praise forever. Beauty for ashes, you find the weak and contrite heart. Shoulder its burdens, carry it into the light. Jesus, Jesus, I need you every moment. I need you here now. This place, Lord, heart sing out your praise forever.
Point I want to invite to the podium, the high table here, the president of the Botanical Society of Nigeria, our revered Professor Adamu A. Alero. A round of applause to him, please.